Hi everyone, can you see me, hear me? Hello. Good evening students. Can you see me, hear me? Hi everyone, good evening. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to talk about the molecular basis of inheritance. Hello, welcome to the session. Okay, so guys, now I know I have seen one question in the chat section that one day, one chapter, this is what you said, then why are you taking the part 2? Because you know that it is one of the most important chapters and it is a very lengthy chapter, right? And it will take me around 12 hours in one go to finish this chapter without compromising the content. Okay, if you want good content, then yes, you have to, right? You have to be patient and you have to read this chapter in detail. Okay, done bache? You cannot complete this chapter in one go. Okay, like if you're thinking that in 3 to 4 hours or in 5 to 8 hours you can finish this chapter, no, not at all, right? You can just revise that chapter in 6 to 7 hours, but if you want to complete it properly, you need 10 to 12 hours, right? At least I am going to take that much time. Okay, because here in this chapter there are many things that students, they do not understand. All the mechanisms, they are important, like the DNA replication, the transcription, translation, genetic code, human genome project, lac operin, even DNA fingerprinting, the structure of DNA, packaging. Each and every topic given here in this chapter is very important. Okay, so here we cannot categorize that yeah, this particular topic is important, this is not, you can leave this, right, and you can read this. No, overall this chapter is very important. But yes, HGP, human genome project, is very important. For sure you will get one question from this topic. Okay, DNA fingerprinting, again very important. Okay, so as per the poll conducted on our Telegram channel, majority of students, they were interested in having the part 2, so that's why I'm conducting it. Okay, yes Arjit, once you understand this chapter, then you can finish it in, even in 2 to 3 hours, then there is no problem at all. Fine, you have to understand this chapter first. And as per your responses in the last class, I was very scared that might be students, they don't even know the ABC of this chapter. Okay, so now without wasting time, let's start the class. And here you people can see we have already started a new batch, that is NEET End Game for the NEET 2024 aspirants, and it is the ultimate crash course, students. We started the classes on 2nd of January and in detail we are completing our syllabus. Okay, so still it's not too late. Be the part of this batch where you have regular classes, where you have proper revision, where you have proper test, where you have proper doubt sessions as per your demand. Okay, so we are going to teach you from scratch, from 0 to advanced. We are going to move in this particular batch. So join the batch now. In the description box there is a link. You have to click on that link and then you have to follow the steps and you will be the part of this batch. Clear, bache? So now one more thing is there. See, we have the scholarship test for NEET UG aspirants, and after clearing this scholarship test you will get 90% of the scholarship, right? 90% of the scholarship you people are going to get. So scan this QR code at just ₹99, right? You will be the part of the scholarship test. Clear, bache? So let's start the class and tell me, what do you know about the, okay, what do you know about the DNA? Whatever you know, just let me know in the chat section, everyone. Whatever you know related to DNA, just let me know in the chat section, and quickly share the link with your friends and tell them ma'am has started the class. And today we are going to start the class with the types of RNA, then transcription, and then genetic code and the translation. So all the topics are important. If you want me to finish this chapter in 4 to 5 hours, right, then I need your support. I want the answers immediately. Yes, everyone, quickly. Tell me. Yes. What do you know about the DNA? And share the session link with your friends and hit the like button, guys. Hit the like button, quick. Quick. It is the genetic material. What else? It has thymine made up of ATGC. It's anti-parallel, follow complementarity. Excellent. DNA has H in second carbon. Seriously, you really need to revise this chapter, bache. When you talk about the DNA, you know that it is a polymer. It is a polymer of nucleotide. That is what our DNA is. Is that clear? Polymer of nucleotide, or in another word, we can say that it is a polynucleotide chain, and that is what we have covered in the last class, bache. So when you talk about it as a polynucleotide chain, means it is made up of nucleotide, means it is having sugar, it is having nitrogenous base, and it is having the phosphate, right? What is a nucleotide? What is a nucleotide? Nucleotide is something having sugar, having nitrogenous base, and having the phosphate. So sugar, nitrogenous base, and phosphate, they all are going to join, bache, and then we are going to get a polynucleotide chain. Okay, so because it is a DNA, so the sugar there is deoxyribose, and here in this sugar, Priya, you need to be very specific. Here in this sugar, at second carbon, at 2' end, there is no oxygen. That is the difference in between the deoxyribose and the ribose sugar. And when you talk about the nitrogenous bases, adenine, guanine, cytosine, thymine, they are present, right? They are present. And which acid do we have here? The phosphoric acid. So that's what we have completed in the last class. Clear, bache? So when it comes to the DNA structure, we know that James Watson, you should know the scientist name, James Watson and Francis Crick. Right, they explained the structure of DNA, the most commonly occurring DNA, that is your bDNA, which is the right-handed DNA. Right, they explained the base pairing and everything. And with the help of the data of Maurice Wilkin and Rosalind Franklin. Right, as per that X-ray diffraction, as per that X-ray crystallography result, right, they explained the James Watson and Francis Crick, they explained the DNA structure. That is what we have covered in the last class. Any doubt, bache? Any doubt? So how the DNA is going to replicate? You know that semi-conservative, right? It is going to follow semi-conservative model of DNA replication. Okay, semi-conservative model of DNA replication in the S phase of interphase. Fine. So who gave the experimental proof for the semi-conservative nature of DNA replication in eukaryotes? Listen to my question carefully. Yes, people, listen to my question carefully. Who gave the experimental proof for the DNA replication in the case of eukaryotes? Yes, in the case of eukaryotes. Excellent, Samriya, Nandini, everyone. It's Tyler and colleagues. Right? Excellent. And you know that they experimented on faba beans. They used the radioactive thymidine. So you have to be very fast in this chapter. Okay? And trust me, you are going to get direct questions, but you need to understand the mechanism first. Fine? And who proposed the model of DNA replication? Everyone here in the class, who proposed the model of DNA replication? Who proposed the model of DNA replication? Quick. Who proposed the model of DNA replication? Yes, it was proposed by, is it Watson and Crick or only Crick? Is it Watson and Crick or only Crick? Do check it. Okay, so then we talked about the DNA replication. Now quick revision of DNA replication. So you know that we discussed that process of DNA replication in detail, and we talk about the prokaryotes. Right, we discuss it in detail, right, in the case of bacteria, in the case of prokaryotes, and this is the main replicating enzyme, that is your DNA polymerase 3. It is the main replicating enzyme. So we also discussed the helicase there, topoisomerase there, DNA polymerase there, DNA ligase there, and the primase there. So tell me quickly in the chat section the function of all the enzymes. Do you know the function of all the enzymes? Yes, do you know the function of all the enzymes? Everyone in the class, do you know the function of all the enzymes? Sure? Sure, sure, sure. Very good, very good, very good. Excellent. So let's talk about the central dogma. Okay. Okay, so let's talk about the central dogma of information. So DNA is going to give us the mRNA, right? And mRNA is going to give us the protein, right? That is what the central dogma of information is. Yes, students, that is what the central dogma of information is. And I told you about the central dogma reverse, also known as terminism. Okay, also known as terminism. So DNA will form the mRNA and mRNA is going to form the protein. DNA is going to form the mRNA, mRNA is going to form the protein, and we know DNA can replicate by its own. So DNA to mRNA, it is known as transcription, and mRNA to protein, it is known as translation, right? It is known as translation. So now, Bache, before understanding the transcription, let me tell you about the RNA, the quick revision of RNA. RNA means ribonucleic acid. What is the meaning of RNA? Yes, Priya, everyone here in the class, what is the meaning of RNA, the full form? You know that it is ribonucleic acid, right? It is ribonucleic acid, it is single stranded, it is having adenine, guanine, cytosine and uracil as the basis. It is having adenine, guanine, cytosine and uracil as the basis. It is single stranded, right, bache? It is single stranded, and bache, this RNA can also act as a genetic material, isn't it? It can also act as a genetic material, and that too in the case of retroviruses, right? That too in the case of retroviruses. Is that clear? Is that clear? Yes, the sugar present here is, this is the point of difference as well, right? Here you have uracil, and when you talk about the sugar present is the ribose sugar. Which sugar is present there? It is the ribose sugar, right? It is the ribose sugar. So now you know that, you know the RNA word, you know that RNA being single stranded, it is very, right, reactive. Moreover, it is having ribose sugar that is having that OH group, right? Because of that also, it is also contributing to its that reactive nature. It is single stranded, that's why it is more reactive, right? It is having uracil also, isn't it? Isn't it? So that's why it was important, because the genetic material should be something which is stable, okay, which should not be very, it should not be very reactive. Okay, it should take part in, it should undergo mutation, but not at a very faster rate. So with the time, with evolution, it was the demand that RNA should evolve into something more stable, and we have the DNA, right? We have the DNA. Clear, bache? So RNA in some retroviruses, like your HIV, right? In that cases, it is still acting like a genetic material, but it is playing other roles as well, like it is acting like a messenger, it is helping in the translation, it is helping in the formation of ribosome. So it is having different functions as well, right? So when you talk about the types of RNA, mainly what are we going to discuss, students? Yes, when we talk about the RNA, when we talk about its types, okay, so we have messenger RNA, we have ribosomal RNA, and we have transfer RNA, isn't it? We have messenger RNA, we have ribosomal RNA, and we have transfer RNA. So first of all, let's talk about it, right? Let's talk about it, and then we are going to discuss about the transcription. Okay, so messenger RNA, ribosomal RNA, and the transfer RNA. Okay, let's start the transcription first, then I'll be teaching you about this mRNA, right? That will be easy for you then. So just write down one thing. mRNA means it is the messenger ribonucleic acid. Okay, rRNA means it is the ribosomal RNA. It is going to form the ribosome structure with the help of some proteins, and this is going to be your transfer RNA, which will act like an adapter, which will help in the protein synthesis, okay, which will help in the protein synthesis. So how that mRNA is going to form, that will be explained with the help of this process known as transcription. So everyone please pay attention, right? Everyone please pay attention. And transcription is very easy if you understand DNA replication. So how many of you understand the DNA replication? Ma'am, HSP sir, ma'am, HSP sir. Kriti bache, HSP sir, he didn't teach bio na? है ना? That's why he's not here. He doesn't know anything related to the transcription. Fine? So quickly tell me everyone, what do you understand by this transcription? Yes, what do you know about transcription? But today's class is very important. Each and every topic that we are going to discuss today is very important. Molecular basis of inheritance. You cannot skip even a single topic from this particular chapter. If you are aiming 360 out of 360 in biology, then you have to complete all the 32 chapters in detail. I hope you all have seen this video. Just a minute. I hope you all have seen this video. See, look at this video section, right? Pankuri ma'am has posted the 25 topics to get good marks in botany, right? I have posted this video 0 to 650 plus marks in your 3 months. Is it possible or not? So even, right, if you follow these strategies, if you follow these videos, right, even the most important topics, the molecular basis of inheritance is the chapter that you cannot leave at any cost. Okay, see, imagine if you will score 350 or 355 marks in the biology, like 300 plus in the biology, even if you are getting 100 marks in physics and chemistry, still you will score 500 plus marks. Okay, still you people are going to get 500 plus marks, right? So please pay attention on such things. Kindly pay attention on such things. Biology is very important for you people, right? And equally you have to score good marks in physics and chemistry if you are really targeting a very good medical college. But at least, right, this is what I'm considering, that minimum you should score minimum 550 marks, right? Minimum 550 marks. This is what you need. And for that, yes, you should score 350 marks in biology. So please pay attention. It is one of the most important chapters and no one is going to explain you this chapter in that detail. So what are we discussing? We are discussing transcription, right? So in the transcription, see, we know that DNA is fully loaded with information, but that's not like that DNA will go to the each and every organelle of cell, right, and will tell that organelle, hey, this is what you have to do, that is what you have to do. No. Here comes the concept of gene expression, right? We know that gene is the basic unit, right? It is the basic unit of inheritance. It is present on the DNA. Right? Let's discuss it in this way. When you talk about the gene is the basic unit of inheritance. It is a basic unit of heredity, right? It is present on that what? On DNA, right? And how a gene is going to express itself? How? By making the proteins. That is how a gene is going to express itself, right? Right, for its expression, what it has to do? It has to form a protein, that is an enzyme which will regulate all the processes, right? Which is going to regulate all the processes. That is what we need. That is what we know. So the very first step is that DNA will form a transcript, right? What DNA is going to do? DNA will not go here and there to tell other organelles, to tell cell that yes, cell, this is what you have to do. The DNA is going to form a copy of the information that DNA is containing. Clear, bache? DNA is going to form the copy of the information that information is present. The information which is present in DNA is going to make the copy of that information, and that copy is basically the messenger RNA. Clear, bache? That copy is basically what? Yes, bache Victor, that copy is basically what? It is the messenger RNA. Is that clear? Is that clear? And overall, let me tell you that messenger RNA, it forms, right? It forms 1 to 5% of total RNA of a cell. Do you know that? It is just 1 to 5% of the total RNA. Usually students, they make this mistake. According to them, the maximum RNA that our cells are having is mRNA. It is not. It is just making 1 to 5% of the total RNA. Clear bache? Clear bache? And let me tell you, it is the least stable RNA. Your mRNA is what? It is the least stable RNA. Okay, it is the least stable RNA, the mRNA. Clear bache? The mRNA. Okay, now you know one more thing. We have, first of all, understand the process, then we will read it from NCERT, right? In this chapter, the most important thing is to understand the mechanism. Okay, so you know that we have two types of cells, right? We have a prokaryotic cell, the bacterial cell. Yes, bache. We have a prokaryotic cell, the bacterial cell, where the genetic material is there in the cytoplasm. And then what do we have? We have the eukaryotic cell, and here in this eukaryotic cell, the genetic material is present in the nucleus, right? This is what we have. Now in the case of prokaryotes, because in the case of prokaryotes, there is no proper nucleus, even the genetic material, even the nucleoid is there in the cytoplasm. And in the prokaryotes, transcription and translation, they occur simultaneously. Transcription and translation, they occur simultaneously. So in the prokaryotic cell, bache, the site of transcription, the site of that copying that information is what? It is the cytoplasm. What is it? It is the cytoplasm. But in the case of eukaryotes, if you talk about the eukaryotic cell, in the case of eukaryotic cell, bache, the transcription, because genetic material is in DNA, so the transcription, genetic material is in the nucleus, so the transcription is also there in the nucleus. Are you getting my point? Yes. Are you getting my point? So this is something very important. In prokaryotic cell, the site is cytoplasm. In eukaryotic cell, the site is nucleus. This is what you need to keep in your mind. Clear bache? Clear bache? Yes. Yes, yes, yes. Understood. Now what is the next point here? That's the first point. Okay. Right, the site of the transcription. Now, what do you think? From where the transcription is going to start? Tell me, what do you think? From where the transcription is going to start? Yes, tell me. Anyone? From where the transcription is going to start? Anyone? The transcription unit. See bache, relate this. When you talk about the DNA replication, what we used to discuss, we used to say na, that DNA replication will start from the origin of replication. Do you remember that? The DNA replication is going to start from some particular sites, right? There should be an origin of replication, right? There should be an origin of replication. We have that ori sites, right? Origin of replication sites are there. Like in the case of prokaryotes, we have, in the case of your E. coli, we have ori C site. Do you remember? This is what we have, ori C site. Right, this is what we have, Priya, ori C site. So DNA replication, it is going to start from the origin of replication. So DNA replication will start from an origin, right? And that site is known as ori C site. Clear, bache? That site is known as what? It is known as ori C site. Isn't it? Isn't it? That is the point that we have. So we used to call that site as even the replicant. Right, replicant, the point from where the replication will start. So you know it very well that in the case of prokaryotes, there is only one origin of replication, so they are monoreplicant. And in the case of eukaryote students, because they have many origins of replication, so they are multi-replicant. They are multi-replicant. Is that clear? Is that clear? That is what we have discussed. So now when it comes to the transcription, please make the notes. And if you are interested in completing that chapter, I want to see the energy in the chat section, guys. Yes, I want to see the energy in the chat section. Quick, I want some fire emojis. It doesn't matter whether we have 100 plus students or less than 100. The main thing is in this chapter, I really don't want to compromise the content. So I am going to take the time right, I am going to take the time which is needed, which is actually needed to complete this chapter. So quickly guys, quickly. Yes, if you are here, make sure that yes, today you are going to learn this chapter, right? You are going to understand this chapter completely. Okay? Very good, very good, very good. So when it comes to the transcription, transcription is going to start, right? And there will be a transcription unit from where we will discuss that, okay, this is the point from where actually the transcription will start, right? The transcription unit. Okay, so now when we talk about this transcription, we need to understand few things. Okay, I'll go one by one. Now in the case of transcription, students, you know one thing, we have a DNA and our DNA is double stranded, isn't it? DNA is what? DNA is double stranded, isn't it? And it is what? It is anti-parallel as well, right? You know that, isn't it? You know that. Tell me. You know that, right? Just a minute. Okay, right? When you talk about the DNA, we know one thing, that DNA is double stranded, right? It is double stranded. And now when you are going to talk about the RNA that we are going to get is, it is going to be single stranded, the mRNA. Now we have the two strands here. So now what do you think? Are we going to copy both the strands or are we going to copy only one strand? Tell me quickly. What are we going to do here? Are we going to copy both the strands of DNA for getting that information, or are we going to focus on only one strand? Only one strand. What is the reason? What is the reason? Why is it so? Right? Why is it so? Let's say this is your DNA. Okay, one strand is having polarity 3', another strand is going to have polarity. One strand is having polarity 3' to 5', the other strand will be having polarity 5' to 3'. Right? The another strand is having the polarity 5' to 3', isn't it? That is what we have. That is what we have. One strand is having polarity 3' to 5'. Another strand is having polarity 5' to 3', isn't it? So we cannot copy both the strands. There are two reasons. The very first is, right, our mRNA is single stranded. If we will copy both the strands, right, if we will copy both the strands are going to give us the different information, right? It is going to give us the different information. Remember that point? I told you clearly that whenever it comes to the synthesis or the polymerization of DNA or RNA, it will always be in 5' to 3' direction. Do you remember this point? It's a very important question. It's a very important question. Yes, bache. Yes, bache. Blue bache, if you are actually grateful, please pay attention here and please focus. Yes, do you know that, students? Right, that whenever it comes to the synthesis is always going to be in 5' to 3' direction. You are polymerizing mRNA or you are polymerizing DNA, it doesn't matter. Synthesis will always be in 5' to 3' direction. Okay, okay. So if you look at this DNA, right, if I need to make the mRNA, I know this is the condition. Synthesis will always be in 5' to 3' direction. So don't you think that I am going to copy the DNA strand having polarity 3' to 5'? Yes, don't you think that I'm going to copy the DNA strand having polarity 3' to 5'? Why? Because it is the one which is acting like the template, right? It is the one which is actually acting like a template strand. So this is another MCQ Leharika, that in the DNA, the strand having polarity 3' to 5' will act like a template. It is the strand that we are going to copy. Let me write it down. Right, that in transcription, the strand this is important, having polarity 3' to 5' is going to be the template strand, isn't it? It is going to be the template strand. That's something very important. Okay, it is something which will be the template. Okay, because complementary to it, we can form our mRNA in 5' to 3' direction, isn't it? Complementary to it, we can form the mRNA in 5' to 3' direction. I'm explaining you this topic in detail, okay, because there are many points here in this topic also which are not clear to students. Okay, that is why I'm going little slow. So please pay attention. Now I'm picking up a random sequence. Let's say here you have A, T, C, G, C, A, T, C, G, whatever it is, G. So now tell me it's a DNA. So what should be here? Indu, if G is there, what should be there on another strand? Quickly tell me the sequence in the chat section. Everyone, quickly tell me the sequence in the chat section. If here you have this sequence, what should be there? What should be there? Yep, in one message write down. In one message write down everything. Obviously it's simple. Here it should be C, T, A, G, C, G, T, A, T, A, G and C. This is the rule of complementarity, isn't it? This is the rule of complementarity and that is what we know, right? That is what we know. So now let's say if I'm going to copy this strand, right, for making my mRNA, what am I going to get on my mRNA? Yes, if let's say this is a strand that I'm going to copy, that what sequence should be there on my mRNA? Tell me, what sequence should be there on my mRNA? Please understand the basics here. Please understand the basics here. What should be there? Excellent, Nandini. You know that in the case of mRNA, you are not going to get thymine. So if G is here, here it should be C. A is here, here it should be U. T is here, here A. G, C, G, U, A, U, A, G and C. That is what we are going to get. That is what we are going to get. Please check it very carefully. That is what we are going to get. Okay, so now let's say just giving you one example. I'm just giving you one example. Let's say I'm copying this particular strand of DNA, right? Forget about the rules. Forget about the rules. Right? Just tell me, bache, no doubt at the place of thymine, uracil will be there. But here when I'm making the mRNA, this mRNA will be complementary to this strand. No. So G complementary to G is C, complementary to A is U, complementary to T is A, complementary to C is G. This is how it is going to be there. This is how it is going to be there. Now focus on this side. Let's say if I have copied this particular strand of the DNA. Here you will be having this, right? Both the sequence are not same, right? Here please pay attention. Both the DNA strands, they are complementary to each other. They are not having similar bases. They are not having identical bases. They are just complementary to each other, right? They are just complementary to each other. That is the point that we have. Fine, that is the point that we have. They are just complementary to each other. So if both the strands of the DNA will get copied, you will get two different types of mRNAs. Two different type of mRNAs means it will give you two different types of information. Why? Why? Because when later on, when this mRNA will be studied, we will pick up 3 bases. You know na, our codon is having 3 bases, right? 3 bases are going to give you, this one codon is going to give you the information about one amino acid. Then there will be the second amino acid. Then there will be the third, and then comes the fourth. Then comes the fourth, isn't it? That is how the amino acids will be added, because we know that 3 bases will give us the one codon. One codon means one amino acid. So if we will take both the mRNAs, we will have different information, we will have different type of protein, and it is not going to work at all, right? It is not going to work at all. First point. That is why it is important to copy only one strand of DNA. Second important point here is, students, that when you look at this mRNA and this mRNA, even they are complementary to each other. They are complementary to each other. If both the strands of the DNA will be copied, these mRNAs, even they will form hydrogen bond and they will become double-stranded. They will form a double-stranded RNA, and that is also of no use, because then we have to put extra energy to open it up, then we have to put extra energy to study both the RNAs. Again, it is of no use. So that is why, students, that is why only one strand of DNA will be copied here, and that strand is known as your template strand. Clear, bache? That strand is known as your template strand. Okay, which will be copied, and template strand is having polarity 3' to 5'. Understood? It is having polarity 3' to 5'. So that is why when we discuss the transcription, we keep one thing in our minds, right? That transcription is going to start from a transcription unit. Fine. The transcription, it is going to start from a transcription unit, and look at this transcription unit. Please look at this transcription unit. This is given in NCERT, right? This is given in NCERT, yes or no? So here, this is just a stretch of DNA. So look at it. One strand is having polarity 3' to 5'. Another strand is having polarity 5' to 3'. So the strand having polarity 3' to 5', it will be your template strand. It is the one which will be copied, right? It is the one which will be copied. And opposite to it, this strand is going to be the coding strand. Clear, bache? This strand is going to be the coding strand. Clear? Everyone in the class, is it clear? So now what is this? This side, this structure is known as promoter, and this structure is known as terminator, and this part in between is what? It is the structural gene. What is it, students? It is the structural gene. Clear? It is the structural gene. Understood? Understood? So when you talk about a transcription unit, so from where the transcription is going to start? Transcription will start from transcription unit, and here you have three things. One is the promoter, one is the structural gene, the gene which will be copied, okay, and then comes the terminator. Then comes the terminator. So promoter, that will promote transcription, structural gene which will be copied to get the information, terminator which is going to terminate the transcription, So here in the structural gene, again you people can see template strand and the coding strand, template strand and the coding strand. So write down these things, bache. You know template strand is having another names also. It is also known as non-coding strand, right? It is also known as non-coding strand. It is also known as antisense strand. Yes, this is the commonly occurring mistake. Okay, sometimes see antisense, anti word is considered as opposite. No, so when we say na antisense strand, so we consider it should be something which will not be copied, but actually it's not like that. Template strand is the antisense strand. Okay, Vishal, template strand is the antisense strand. So template, non-coding or antisense strand, it is also known as minus strand or the negative strand. Understood? And when you talk about your coding strand is also known as, please write down, your coding strand is also known as non-template strand. It is also known as sense strand. It is also known as positive or the plus strand. Understood? Positive or the plus strand. Clear, bache? Clear, clear, clear? Yes, template non-coding antisense, coding non-template sense, or the positive strand. Is that clear? Is that clear? Can I ask you one question? Can I ask you one question? Basically 2 questions. Yes, everyone? Okay. So my first question here is that out of, let's say this is also, okay, and yeah, the function of promoter and terminator I told you, but we will discuss each and everything written here in detail, but as of now you should mark the transcription unit. Okay, so now my point is, when you look at this DNA, which one is the template strand here? Which one is the template strand here? Yes, which one is the template strand here? Please have a look. Of course, this one, right? This is our template. It will be copied. It is in 3' to 5', so complementary to it, you will get mRNA in 5' to 3' direction. But you look at the bases here. Okay, look at the bases here. It is now C, U, A, G, C, G, U, A, U, A, G, C. These are the bases that we have. Priya, Devitha, Niharika, Chetu, Anjani, Vishal. This is what we have, yes or no? This is what we have, yes or no? Yes or no? So when you look at the bases of the mRNA, try to compare it with the coding strand. Yes, try to compare it with the coding strand and tell me, what do you observe? Please tell me, what do you observe? Same code, है ना? Same code. This mRNA is having the same genetic information. The only difference is that at the place of thymine, you are going to see uracil in mRNA. You are going to see uracil in mRNA, isn't it? Isn't it? So that is why you call this strand as the coding strand, because the information written on coding strand will be it will be the same information on the mRNA. The only difference is at the place of thymine, uracil will be there. At the place of thymine, uracil will be there. That is the only difference, people. Okay, that is the only difference here. Understood? Yes. Is it clear? So let's say if in the paper there is a question, if you will solve PYQs, you are going to get the questions like this, right? There is a question, they are giving you some, right? Let's say they are giving you what? They are telling you, they are giving you a DNA stretch and they are asking you what should be there on mRNA. So you have to be very smart. Just pay attention on polarity. It should be like this. So whatever is written on coding strand, just put that directly here. At the place of thymine, just use the uracil and you will get your answer. It will save your few seconds. That you can use for another question. Understood? Understood? So this question can be asked in different ways. So you people should know about it. Is it clear? Is it clear? Any doubt? No? Sure? That's why coding strand is that's why it is named like this, coding strand, because it is having the same information. It is having the same information like what? Like the mRNA. Understood? Okay. So next question, my next question is, this is one part. Now when you talk about a gene, it is an NCERT, right? So when you talk about a gene, what is a gene? It's a basic unit of inheritance. What is a gene? A gene is a basic unit of inheritance. That is what a gene is, right? Functional unit, yeah, basic unit, whatever it is, right? So gene is a functional unit of inheritance, whatever you want to call it. Okay, functional unit or the basic unit. Chalo, let me write down properly. Okay, so it is the functional unit of inheritance. That is what we have, functional unit of inheritance. Now I have a point here. When we talk about the gene, right, ultimately DNA is going to give us the information. Ultimately, the gene present on DNA is going to express itself. That is what you know. That is what you know. Bache, you are again and again asking, Satish Bache, which is parental strand. Bache, what is the meaning of a parental strand? You can relate it with the replication. When you copy a DNA, right, you open up both the strands. Both are acting like a template. They are the parental strand. When the new daughter DNA will form, it will have one parental strand, one newly synthesized strand. Parental strand which is coming from the parents, right, as such which is coming from the parents. So why are you confusing it here in the transcription? Okay, so gene is what I just said. Gene is the functional unit of inheritance. So ultimately, gene is going to express itself, right? Ultimately, a gene is going to express itself, student, and gene is the one which will give you the protein. But, is gene is just going to give us the protein? Are we only going to get protein from the gene? Of course, it's a big no, right? From the DNA, from that gene, even it will form ribosomal RNA, right? Even it will form transfer RNA. And moreover, these segments of DNA, right, they are playing role in replication also. They are playing role in crossing over also, right? They are playing role in mutation also, right? There are different things, different things. So we cannot simply say, if we are saying gene, we cannot simply understand that it is going to give us the protein. Am I right or wrong? Yes, am I right or wrong? Right, can we define gene in terms of DNA sequence? Because DNA sequence is coding for tRNA also, rRNA also, right? Right. Isn't it? Can we say that gene is a DNA segment? Can we just simply say that? Yes, it is in NCERT, right? Don't you think it is difficult to define gene in terms of DNA sequence, because it is also giving us, DNA is also giving us ribosomal RNA, it is also giving us tRNA, it is also taking part in replication. Again, I'm repeating, taking part in crossing over. There are many things. So how can we define the gene, right? We cannot literally just say that gene is something which is giving us we cannot say DNA segment is something which is giving us mRNA. No, we cannot take it like this. We cannot say that DNA segment is something which is giving us, when we are using the word DNA segment, we cannot assume that it is only giving us protein. Right, Victor? We cannot assume that DNA segment means it is only giving us protein. No, there can be a confusion. So we use the word cistron. Right, we use the word cistron. So what is cistron? If cistron word is written, it is telling us that it is the DNA segment which is giving us polypeptide, isn't it? It is the DNA segment which is giving us polypeptide, right, bache? Right, bache? Because the information of protein, the DNA overall, it is having information for different things, but the information for protein is present on that part of DNA which is giving us mRNA. है ना? If DNA is giving us mRNA, means that mRNA will give us the protein. Right. So cistron is the word that we are going to use. So it is the segment of DNA, right? What is cistron? It is the segment of DNA which gives the information for the synthesis of a single polypeptide chain. Isn't it? It is something, cistron, it is something which is coding for polypeptide chain. It is that part of DNA segment which will form polypeptide chain. Why am I using the word polypeptide? It's simple now. Proteins are made up of what? They are made up of amino acid. Many amino acids, they will join together with the help of peptide bond. Right? Many amino acids, they will join together with the help of peptide bonds. So many peptide bonds are together. It's like a polymer here, na? So proteins, can we use the word polypeptide? Of course, yes. And in general, when we use the word protein, right? Protein means it's not mandatory that protein is only made up of one polypeptide chain. No, bache. No, it's not mandatory that protein is only made up of one polypeptide chain. No, protein can have more than one polypeptide chain, like your quaternary protein, hemoglobin, having 4 polypeptide chains. Okay, so cistron is something which is coding for one polypeptide chain. Understood? Cistron is something, it is a segment of DNA which contains signal for synthesis of only one polypeptide chain. Understood? That is the cistron, right? It contains the signal for the synthesis of only one polypeptide chain. Understood? Understood? Now one more point here. When you talk about the prokaryotes, okay, when you talk about the prokaryotes and the eukaryotes, this is important, right? It's a previous year question, so please pay attention. So P for prokaryote, P for primitive, right? P for prokaryote, P for primitive. You know this thing, right? So when you talk about the prokaryotes, they are polycistronic, okay, they are polycistronic, and your eukaryotes, they are monocistronic, students. Isn't it? Prokaryotes, they are polycistronic, eukaryotes, they are monocistronic. So it means in the case of prokaryotes, right, your structural gene, it is having the signal for the synthesis of more than one polypeptide chain. Polycistron means, right, that one unit, or you can say that one transcription unit can have more than one structural genes. It can have more than one structural genes, means more than one polypeptide chains can be obtained. But in the case of eukaryotes, it is only monocistronic. So it will give you the, it is having the signal for only one polypeptide chain. So it means one RNA polymerase can give you only one polypeptide, but here it will be more than one. Understood? This is important. This is very important. In the NEET paper, they can directly ask you, out of the prokaryote and eukaryote, which one is polycistronic, which one is monocistronic? Definitely in the MCQ format. Or the another way to ask this question is, they can give you example. Let's say yeast, right? Chlamydomonas, okay, and they are telling you about the your cycon, and one another example you can pick up. So they'll ask you, out of the following, in which one is monocistronic, which one is polycistronic. So obviously prokaryotes are poly and eukaryotes are monocistronic. Clear? Right? Eukaryotes are monocistronic. That is what you need to remember. So any doubt here? Any doubt here? Monocistron, polycistron, clear? And is it clear that why do we pick up only one strand of DNA to copy the information? Why do we pick up only one strand of DNA to copy the information? Okay, so now let's start the process in detail. Let's discuss the process of transcription in detail, okay? So are you ready for it? Yes, are you ready for it? So let's revise it from NCERT a bit. Let's include the NCERT as well. So process of copying genetic information from one strand of DNA into RNA is the transcription. Right, Leharika? The process of copying genetic information from one strand of DNA into RNA is termed as transcription. So the principle of complementarity governs the process of transcription. Again, MCQ can come from this statement, that which principle governs the process of transcription. So try to understand the language of NCERT as well. Okay, so it is the rule of, principle of complementarity. What is it, people? Rule or the principle of complementarity. Clear, bache? So except the adenosine, the only difference is, except the adenosine complements, except that adenosine is going to form with, going to pair with the uridine. Yeah, complementary to adenosine will be the uridine, not the thymine. Okay, done, bache? So now what are they saying? That in the process of replication, you know that the complete set of DNA will be duplicated, but in transcription, only a segment of DNA. And you know that that segment of DNA which will be making mRNA, it is known as your cistron, right? It is the cistron. Clear, bache? Clear, bache? And you know only one strand of DNA will be copied into the RNA. Is that clear? Is that clear? Right? So I already told you about this thing, that why both the strands are not copied during transcription. Can you explain me that in the chat section, quick, everyone? And guys, quickly hit the like button. The energy should be high. Your energy should be high. So everyone, just quickly let me know about it. You know the answer, you just need to tell me in the chat section. Yes, you know the answer, you have to tell me that answer in the chat section, that why both the strands are not copied during transcription. Whatever you know, type it quickly. Yes, exactly. So see, if both the strand will act as a template, they will code for RNA with different sequences, right? So in turn, if they code for proteins, the sequence of amino acid in the proteins will be different. So basically it will give us different information, right? This would complicate the genetic information transfer machinery. Secondly, bache, the two RNA molecules, if produced simultaneously, they will be complementary to each other. They will form a double-stranded RNA. This would prevent RNA from being translated into protein, and this is something which we will discuss in RNA interference. Okay, Samiksha, this is something that we are going to discuss in RNA interference. Then when it comes to the transcription unit, that's the diagram that we have. Okay, that is the topic that we have, right? So look at this point. Please look at this point. This is your structural gene having one template strand, having one coding strand. So in the NCERT itself, please mention the another names. Do not miss this point. Template strand is also known as a non-coding strand. It is also known as antisense strand. It is also known as minus strand. And the coding strand is also known as non-template strand. It is also known as sense strand. It is also known as positive strand. Understood? Understood? And here you have promoter, you have terminator, right? And this is actually going to be the start site from where the transcription will start, right? One more thing that you people miss, if you will look at the arrow here, the arrow is in this direction, 5' to 3'. Do you know why? It is telling us that synthesis is always in this direction, 5' to 3'. Like this is the 5', this is the 3'. This direction of arrow is telling us about the synthesis. So do not get confused here. The arrow will be in this direction, 5' to 3'. Okay, the 5' to 3'. So these are the minor details that we people should not miss. Clear? Clear? So you know that the two strands are the structural units. So since two strands have opposite polarity, right? So your DNA dependent RNA polymerase will catalyze the polymerization in one direction only, that is 5' to 3', and the strand having polarity 3' to 5', it will act like a template. Important MCQ. Important MCQ. Understood? Understood? Clear? So see in the NCERT, look at the focus here, bache. This is the language of NCERT. So the other strand which has the polarity 5' to 3', it means they are talking about coding strand. So its sequence is same as RNA, except thymine at the place of uracil is displayed during transcription. Understood? Sure, bache? Sure? Okay. Now there is one point. Let me elaborate that. Strangely, this strand which doesn't code for anything is referred to as coding strand. So if you don't know the process in detail, if you do not understand the mechanism, bache, might be you will confuse that thing, right? You will say, Arey, if it is the coding strand, it is something which will code. No, no, no. Actually, it is the template strand. It is the template strand that will code. Understood? Understood? Now, all the reference points while defining a transcription unit is made with coding strand, right? Let me tell you the meaning here. Now let's say, first of all, focus on two words. Many teachers, they make the mistake here. I have seen many teachers explaining this thing in a different way, right? Or I'll say in an inappropriate way, okay? You please understand. Let's say you are considering this as the start point. Okay, this is something which you people are considering as a start point. It is something from where a process is going to start. So my dear kids, if you are moving towards the upside, like this side, if this is the side, you will use the word upstream, right? If this is the side, you will use the word downstream. Are you getting it? Are you getting it? Let's say this is the reference point. If you are moving towards this direction, you are going upward. This is how you can keep it in your mind. This is upstream, this is downstream. Now in NCERT, the topics, these transcription, translation, they are given at a very basic level, right? When you study such topics in your higher grades, then you get to know about the detail, right? Then we use these terminology, downstream, upstream, to tell the location of the bases as well. Just say 10 base pairs downstream, 10 base pairs upstream, means from the start points 10 base pairs upstream. That is how we use it, Clear? Clear? But in your NCERT it is given, very basic concept is given, right? So let's understand in that way. So if this is the start point, students, upstream, this is the downstream. Now come to this side, as I said, right, जैसे coding strand is like a crybaby, ठीक है, coding strand is like यार that's not fair, you are not even coding me. Okay, fine, you gave me the name coding strand, you are not even using my genetic information. Obviously on mRNA the information is same like coding strand, but ठीक है, coding strand is upset, and coding strand is like कि I want you to make the references, right, by considering me. I should be the reference point. What is the meaning of this? Like, let's say it is a very repeated question in the paper. Let's say if you are taking coding strand as a reference, then what will you say? The promoter is present at 5' end or at the 3' end? The promoter is present at the 5' end or at the 3' end? I am telling you, students, if you are taking the reference of the coding strand, I clearly mentioned this, and in NCERT it is clearly written that generally the references are made by the polarities and the directions or the points, they are explained by taking the reference of coding strand, right? So if this is the coding strand, clear bache, if you are talking about the promoter, it is present at its 5' end, right? Obviously upstream, it's upstream, it is present upstream and at 5' end, and the terminator is present at 3' end. But if they are taking the template strand as a point of reference, then it will be at 3' end and 5' end. So in your question, in your final paper, if you get any question like this, please make sure that you read properly, right? Properly there, it should be mentioned, right? I have seen few MCQs on the basis of this concept, and in that MCQs they clearly mention the reference point, right? So if we are taking the reference of coding strand, then it is at 5' end, terminator is at 3' end. If you are taking the template strand as a reference point, then promoter is at 3' end, terminator is at 5' end. So please do not confuse. And let's say if there is a question where they did not mention anything, any reference point, okay, then in that case consider the coding strand as a reference point. Okay, Gangana, if they didn't mention anything, any reference point, then you should consider coding strand as a reference point, because in NCERT it is clearly mentioned, right? It is clearly mentioned that the reference of coding strand will be taken. Understood? Sure? Sure? Okay? Just a minute. Done? Is it clear? Sure? Sure? Any doubt? So start point, upstream, downstream, this part clear. So all the reference point while defining a transcription unit is made with the coding strand. Okay, so they have given you one hypothetical sequence, template and coding strand, right? And they are asking you, you know, which one is present at what side. So you should know about it. So see, the promoter and terminator flanks the structural gene in a transcription unit. So promoter is towards the 5' end upstream. So what students used to do, they just focus on this thing, that promoter is located towards 5' end, means upstream. Upstream, as again I'm telling you, why upstream? See, this side will be upstream, this side will be downstream. That's it, that's all. And do not just focus on this thing, that promoter is said to be located towards 5' end. Here they are taking, here the reference point is your coding strand. Okay, so if I will change the reference point, the polarity will change. It's like this. Like, let's say now I'm facing the camera. Okay, so I'm saying the door is towards my right side, the almirah is towards my left side. Okay, now let's say if I'll be like this, then I'll say the door is towards my right side, almirah is towards my right side, door is towards my left side. It's like that. It is the meaning of taking a reference point, right? Let's say you are explaining direction to someone. If you are facing a shop, right, shop, then accordingly you'll tell, है ना? Let's say if you are not facing that shop, right, you are standing in opposite direction, then accordingly you will tell me, right? Accordingly you will tell me. So please keep it in your mind. If here they are mentioning, they are taking reference point as the coding strand. Any doubt? Any doubt? See, here also it is explained. Okay, okay, so now let's understand the mechanism in detail. What's it? Are you ready? Yes, are you ready to understand the mechanism in detail? It's very simple. Now in the, yes, one more thing. Okay, now when it comes to the transcription, again in the prokaryotes and in eukaryotes differences are there. In prokaryotes, the site for the transcription is, yes bache, the site for the transcription is cytoplasm. You know it very well, isn't it? You know it very well. And when it comes to the enzyme that is going to make your mRNA, that will be your RNA polymerase. That is going to be your RNA polymerase. And students, why do we say RNA? See, it is RNA polymerase. It is also known as DNA dependent RNA polymerase. What is it? It is also known as DNA dependent RNA polymerase. Why DNA dependent? Because it is using DNA as a template for making RNA. That is why it is DNA dependent RNA polymerase. If somewhere it is written like this, na RNA dependent RNA polymerase, it is incorrect here, because here this enzyme is using the reference of DNA. This enzyme is using the reference of DNA. Clear? Now obviously we know about the transcription point, right bache? Unit. So we have something called promoter. So promoter means to promote something. So obviously it is something which will promote the transcription. Am I right? It is something which will promote the transcription, which will help in the initiation of transcription. Students, when we discuss these processes, transcription, translation, we divide it into 3 steps. Okay, we divide it into 3 steps. That 3 steps are initiation, means the starting of that process. Then elongation, means further bases will be added, right? And then the termination, that how that process will stop. Clear? So here for the initiation, for the starting of that particular, process, the promoter site is going to play the role, because promoter site is the one which will promote the transcription. It will provide a site for the enzyme that will make the mRNA. Clear bache? Clear? So now if I have to, Priya, if I have to copy the information, I need to open up my DNA. I need to open my DNA, understand this process in detail, then we'll be fast. I have to open my DNA. So here the question is, which enzyme is going to open up the DNA double helix in transcription? It was a previous year question in NEET 2020. And many times, right, we make mistake in such questions, right? Even when for the first time, even when I was solving the paper, I was just like, which enzyme is going to open up the DNA helix? What came in our mind? Helicase. But it is incorrect. It is incorrect. I didn't read the question carefully. In NEET 2020, the question was, which enzyme opens up the DNA double helix in transcription. So when it is the transcription, it is going to be the RNA polymerase. So it is the one in transcription, it is the one that opens up DNA double helix. Okay, this is the one which is going to open up the DNA double helix. Understood? Understood? Yes, bache, understood? Sure? Sure. Right? So where this RNA polymerase is going to bind? It will bind to the promoter site. It is the promoter site that will help for this process. So bache, because it is in the case of prokaryotes we are discussing, right? In the case of prokaryotes we are discussing this process, right? We are discussing this process in the case of prokaryotes. So it's very simple, right? Remember this point. In prokaryotes, there are certain factors. They are not in eukaryotes. So in prokaryotes there is one factor known as sigma factor. Which factor it is? Sigma factor, right? Sigma factor. S for sigma, S for start. So it is the one which will help in the initiation of the transcription. It is the one which will help RNA polymerase to bind to the promoter site. Sigma factor helps RNA polymerase to bind to promoter site. Clear, bache? To bind to promoter site. So S for start. So it will help in the start. Is that clear? It will help in the start, sigma factor. Now look at this. You know this is how our DNA is. 3' to 5', 5' to 3'. And here you have the promoter site. Am I right? Because we are talking about the transcription, that's why I'm taking the example of promoter. And I told you that here it is going to be the start side. You know na? It is going to be the start side, Leharika. Yes, bache? Kanagaraj. It is the start side, this white colored. It is the start side. Now here in the promoter, right? Like you can say that 10 bases upstream, near about 10 bases upstream to this start site, in the promoter region, you will get a AT rich sequence. Listen to me, this is not given in NCERT, this is important, right? The point that I'm discussing right now, it is not given in NCERT, but this is important. So here in this, right, in this DNA, we are talking about prokaryotes. So in prokaryotes, right, in the promoter region, there is a AT rich sequence. What is there? AT rich sequence means it is having adenine thymine. So basically this is the sequence TATAAT, right? This sequence is present and it is known as Tata box. And in the case of prokaryotes, we call it as Pribnow box, right? In the case of prokaryotes, it is known as Pribnow box. The Tata box is there, right? How many bases are there? 6 bases. So this is AT rich sequence which is present in promoter. And why this AT rich sequence is important? You know the bonding in between adenine thymine. See, it is present on one strand of DNA. Complementary to it, you will be having ATA, TTA. So basically, it's easy to break the hydrogen, 2 hydrogen bonds, then the 3 hydrogen bonds, right? That is why this is the point actually. This Pribnow box, it is present in promoter. It is going to provide the site for the joining of RNA polymerase. Right, it is going to, Nandini, provide the site for the joining of RNA polymerase. So sigma factor will bring the RNA polymerase here, will make sure that RNA polymerase joins here. Clear, bache? So which factor is going to play the role here, Nandini, Leharika? The sigma factor will bring the RNA polymerase to this promoter site, will make sure it joins here. Once RNA polymerase joins here, once it starts the mRNA formation, then sigma factor will leave. So sigma factor is the part of initiation but not the elongation. Sigma factor is the part of initiation but not the elongation. Is that clear? Again, I'm repeating, sigma factor is the part of initiation. It is not the part of elongation. It will just help the RNA polymerase to join there and to start the process. Then it will say bye to that RNA polymerase. Is that clear? And one extra info here. that in eukaryotes, also there is such, do you know that even in eukaryotes you are having that Tata box? Clear? You are having that Tata box, and that Tata box is TA. It is having 7 bases, and that is known as Goldberg-Hugeness box. That is known as Goldberg-Hugeness box, right? So in prokaryotes it is known as Pribnow box, okay, and in eukaryotes it is known as Goldberg-Hugeness box. Understood? So in eukaryotes, this is the Goldberg-Hugeness box. Understood? Okay. So at the point RNA polymerase enzymes break hydrogen bonds between 2 DNA strands, it will separate that DNA strands, right? It is going to separate that DNA strands. Understood? Sure, bache? So transcription will also proceed in 5' to 3' bases, right? It will also proceed in 5' to 3' bases. Rest you know what is going to happen in the cytoplasm. All that UTPs, right, ATPs, CTPs, GTPs are present, and same like DNA replication, they will also serve dual purpose, right? These ribonucleoside triphosphates, bache, these ribonucleoside triphosphates, they will be picked up, they will be added to that mRNA, right? They will release their 2 pyrophosphates, and energy, the energy that will be released will be used for polymerization. Okay. So that is the point that you need to remember. So this is about the initiation of the transcription in the case of prokaryotes. Is that clear? So actually it is the start site from where that, right, from where that mRNA formation will start. Clear, bache? Clear? Now when it comes to the termination, for termination there is a rho factor. Rho in Hindi means to stop, rokna, right, to stop. So rho factor, it is for the termination. Okay, so once that rho factor will come, it will terminate the transcription. Sometimes transcription can be terminated in rho independent manner, right? Sometimes rho factor is also not there. In that case, a loop like structure will form and the transcription will stop. Okay, so this is what you need to remember in the case of, this is what you people need to remember in the case of transcription in bacteria. So look at this diagram: promoter, RNA polymerase, and the sigma factor means for the start. So it is not playing any role in elongation, it will just help the process, help this process to start. Okay, then rho factor, when rho factor will come, it is indicating termination. Okay, so RNA will get separated, rho factor will take away the RNA polymerase. So yes, the diagrams are important. Understood? The diagrams, they are important. Okay. So this is all about the transcription in, yes, this is all about the transcription in prokaryotes. So now, bache, when it comes to the eukaryotes, it is not little bit, but yeah, it is different. It is not ditto same, it is different. Can you tell me the differences? Yes. Can you just tell me the differences? Yes. Can you just tell me the differences? Be quick, please. Please. Bache, how can you say capping and all? Like, try to explain it. Please try to explain it. Excellent. So in eukaryotes, first of all, the very first point is, as you said, it will be there in the nucleus. That is the site. That is the site of transcription, and that is going to be the nucleus. Very good. And now bache, in the case of eukaryotes na, there is a concept of split genes. Do you know about split genes? This is important. In eukaryotic transcription, there is no sigma and rho factor. Do you know that? But in the case of eukaryotic transcription, there are other factors like transcription factor D, transcription factor 2D, transcription factor A, B, G, H, E, like this, right? But that is not the part of our syllabus, so we are not going to discuss about this. Clear, bache? But this is what you have to remember, that in eukaryotes we are not going to talk about the sigma and the rho factor. This is important. Okay, it is important for NEET. It is something very important for NEET. Okay, and now let's talk about the split genes. So in the case of eukaryotes, we have the split genes, right? The concept of split gene is very interesting. It is having evolutionary significance. Actually, here in case of eukaryotes, the DNA segment is not always giving us the information for protein synthesis. It is having the non-coding part, and it is also having the coding part. Yes or no? It is having the coding part and non-coding part. Now ma'am, what is the meaning of coding and the non-coding part? Hai na? This question can come in your mind, that ma'am, what is the meaning of coding part and the non-coding part? It's very simple. Non-coding part is not going to give you the information for the protein synthesis, and the coding part is going to give you the information for the formation of protein. So non-coding part is known as intron, and the coding part is known as exon. So in the case of eukaryotes, we talk about the introns and the exons. Introns non-coding, exons coding. Introns non-coding, exons coding. This is what you have to discuss. This is what you have to keep in your mind. Okay, so obviously if in the eukaryotes you have split genes, right? You have split genes, but genes for histones, they are not split genes, but majority of genes of eukaryotes, they are split genes. Clear bache Niharika? They are split genes. Majority of genes, okay, majority of genes, they are what? They are the split genes. So obviously if the DNA segment is non-coding part or the coding part, of course the mRNA, right? Of course the mRNA that we are going to get from that, mRNA that we are going to get from that DNA, it will be the same. Yes, it is also going to be the same. It is not going to be the something which will directly give us the information about proteins. So here also you will see one thing, right? You will see that in mRNA also exon introns, they are present, right bache? So that mRNA, right, that mRNA is not fully functional, right? Isn't it? That mRNA is not fully functional. So here you will see exon introns in between. Can you see that? Here you will see exon introns in between. Understood? Understood? So in the case of eukaryotes, in the nucleus, right, when this transcription is going on, right, the mRNA that you people are going to get, it is not the mature mRNA. Instead of that, this mRNA is known as the immature mRNA. Okay, this mRNA is known as the immature mRNA, or we also call it as heterogeneous nuclear RNA, or we can also call it as hnRNA. Are you getting it? Yes bache, are you getting it? So it is known as hnRNA, or it is known as heterogeneous nuclear RNA, right? It is the unstable RNA. What type of RNA it is? It is the unstable RNA, right? Why? Because it is having coded and the non-coding parts. So if we want to make it mature, we have to remove the introns. Is that clear? We have to remove the intron. Is that clear? Is that clear? This point clear? Now the another difference is that in eukaryotes, right bache, in eukaryotes you just do not have the single RNA polymerase. In eukaryotes, we have RNA, three different types of RNA polymerases are there. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3 is there. Are you getting my point? RNA polymerase 1, 2 and 3. That's what we have. Okay, that's what we have. So when it is the RNA polymerase 2, it is the one which will give you mRNA, or you can say that heterogeneous nuclear RNA. Okay, immature mRNA. This mRNA polymerase 2. Are you getting it? Now 3 means 3, T. So it will form tRNA. It not just tRNA, also form snRNA. Small nuclear RNA, right? Small nuclear RNA. Your RNA polymerase 1 will give you ribosomal RNA. So this information is important. Clear, bache? This information is important. Clear? So immature mRNA, unstable it is, also known as heterogeneous nuclear, also known as hnRNA, or you can simply call this hnRNA as precursor of mRNA. Right? You can also call it as what? Precursor of mRNA. This information is important. Now, please add one more point here. Which ribosomal RNAs are formed with this RNA polymerase 1? It is 28S ribosomal RNA. It is 18S ribosomal RNA. And it is 5.8S ribosomal RNA. Bache, this is what I am assuming that you know what is S, sedimentation coefficient, Swedberg unit. What is it? Sedimentation coefficient, Swedberg unit, right? So these are the ribosomal RNAs which will form your ribosome. So what you need to remember here, that we have 5S ribosomal RNA also, but that 5S ribosomal RNA, that will be synthesized by RNA polymerase 3, and that too, like they all are synthesized in nucleolus. You remember the nucleolus? But this one will be there in the nucleoplasm, outside the nucleolus. Okay, outside the nucleolus. Clear, bache? Do you remember that point? Yes, people. Do you remember that point? That nucleolus is the ribosome factory. Why? Because it forms the ribosomal RNAs, right? But here, this in nucleolus, 28S ribosomal RNA, 18S ribosomal RNA and 5.8S ribosomal RNA will form. And when it comes to 5S ribosomal RNA, it will be formed by RNA polymerase 3 outside the nucleolus. Basically in nucleus, but outside the nucleolus. Done, Niharika? Is it clear? Sure? Yes. So these are the differences in prokaryotes and eukaryotes. So in eukaryotes, no sigma, no rho factor. Site is your nucleus, right? Split gene concept is there. So if we need the mature RNA, we need to remove the introns. We have to join the exons, right? So in the case of eukaryotes, remember one thing. This is a nucleus, right? We have to send our immature RNA to the cytoplasm, because bache, no matter what type of cell is, the translation will always be there in the cytoplasm. Are you getting it? Right? The translation is always there in the cytoplasm. Are you getting it? The translation is always there in the cytoplasm. Clear? So basically I have to send my immature RNA, right? I have to send my immature RNA, mRNA to the cytoplasm. I cannot send it like this. Let's say if your mothers, if they are going to, right, Mushraf bache, if your mothers are going to send you to a college, right? So obviously they are not just going to send you like this. Okay, fine, go. No, you are going to get a lecture, right? All your relatives are going to tell you this is what you have to do, that is what you have to avoid. Let's say if you are going to a place where the temperature is cold, they will give you all that cap, they will give you all that woolen stuff, they will teach you that is how you have to behave. They will tell you about the people, right? About the variety of people that you are going to get and how you need to protect yourself, right? So same is the case here. How can I send my immature mRNA directly to the cytoplasm? I have to process it, right? I need to do what? I need to process it. Yes, people, what I need to do? I need to process it. So I need to convert hnRNA which is unstable, right? I need to convert hnRNA into the stable mRNA, right? Into my stable mRNA, and that will be done with the help of RNA processing, and that is what you need to remember. Okay, that will be done how? With the help of RNA processing, right bache? Right bache? Right? RNA processing. So RNA processing will be only in eukaryotes, not in prokaryotes. Understood? It will only be in eukaryotes, not in prokaryotes. Understood? Understood? So in the RNA processing, what will be done? Yes, bache. In the RNA processing, what will be done? Let me tell you. Okay. Tell me, this chapter is interesting now. If you understand this chapter, there is nothing to cram. Right? If you know the concept, there is nothing to cram. And trust me, you will get very good marks in your final paper as well. Okay? You're actually going to get very good marks in your final paper as well. Right? Try to understand the concepts here. Fine, try to understand the concepts here. So in RNA processing, we'll be talking about the capping, we'll be talking about the tailing, and we will be talking about the splicing. We will be talking about the capping, tailing, and splicing. Okay, now let's start with the splicing. Splicing means removal of introns. What is the meaning of splicing, bache? Splicing means removal of introns. Yes, students, what is the meaning of splicing? Everyone in the class, quickly tell me. It means removal of introns. So how can that be done? Wait. So basically I need to, right, basically let's say these are the introns. So I need to remove the introns. I need to remove the introns and I have to join the exons all together. What I need to do here? I need to remove the introns and I need to join the exons all together. And bache, that will be achieved with the help of snRNAs and some proteins. Right? snRNA means small nuclear RNAs, right? Small nuclear RNAs, of course they are present in nucleus. So when these small RNA, nuclear RNAs, they are going to join with some proteins, right? When they are having some proteins, what do they have? Let's say small nuclear RNAs along with some proteins, they make bache snRNPs. Please pay attention. They are going to make snRNPs, means small nuclear ribonucleoprotein. Small nuclear ribonucleoprotein. That is what they are going to make. Small nuclear ribonucleoprotein. They are, bache, they are pronounced as NERPs. What are they? Right, just say in SNPs. Right, we will also discuss SNPs. SNPs means single nucleotide polymorphism. We call it as SNPs, but they are snRNPs, small nuclear ribonuclear protein, and they are pronounced as SNRPs. Right, they are pronounced as SNRPs. Clear, bache? Clear, bache? So when small nuclear RNA with some proteins, they form snRNPs. Further, these snRNPs, these SNRPs, with some proteins, they form some structures. You know what we used to call it? What we call it? It is spliceosome. It is spliceosome. I know you must be thinking that this word is not in NCERT. Why are you telling this us? You can solve a previous year question. You are going to find this word. Spliceosome is nothing. It is just a group of these SNRs and some proteins. What is a spliceosome? What is a spliceosome? It's a group of some RNPs, means your SNRs with some proteins. They here to remove the introns. Right, they are here to remove the introns, right bache, and then finally the exons will be joined. This is your splicing, right? This is your splicing. So splicing, or you can say that RNA processing, will only be there in the case of eukaryotes. Clear? Clear? So RNA processing is only there in the case of eukaryotes, remember. So yes, they can ask you the question like this. They'll give you the example. Let's say you have the protozoans, you have yeast, okay, you have protozoans, you have yeast, you have euglena, right? You have yeast, you have euglena, you have your protozoan, randomly I'm taking this word, and you have your E. coli. Out of the following, which one, in which organism spliceosomes are absent? Tell me. Spliceosomes are absent. Out of the following, in which organism spliceosome is absent? The options are yeast, protozoa, your euglena and E. coli. E. coli, why? Why E. coli? Because E. coli is a bacteria. Why not yeast? Yeast is a fungi. It's a eukaryotic cell. It's a eukaryotic cell. So this is what you need to know, right? Spliceosome. Wait, I'll show you the image if I'll get the one. See. So these nerves, okay, these SNRs, they will come here, right? They will form a kind of structure like this. Like we also discussed lariat formation, this and that, but that is not the part of your syllabus. See, all of them will come, they will remove the introns, they will break the bonds here, it will be removed and the exons will be joined. Clear, bache? Right? So basically 5 snRNPs, they are needed to bind to the intron, they will remove it, only exons will be there. Clear, bache? Understood? A loop-like structure will form and that's how they will remove. Again, you can see these SNRs are coming, they will hold this intron, they will break it, they will cut it, right? The spliceosome is nothing, it is just the group of that SNRs with other proteins, and RNA lariat will be removed. So this is about the splicing, and this is your spliceosome, nothing but just a group of SNRs with some proteins, or just a group of SNRs, that's what you can say. Understood? Sure? Sure, so this is splicing. So now what about the capping and tailing? Okay, what about the capping and tailing? So when it comes to the capping, bache, capping means basically it will be done at 5' end, right? So to that 5' end, 7-methyl guanosine, write down, 7-methyl guanosine triphosphate, 7-methyl guanosine triphosphate will be added to that 5' end. Are you getting it? What will be done? 7-methyl guanosine triphosphate will be added to the 5' end, right? Right, it will be added to the 5' end. But it is an unusual base. Can you tell me why? It is an unusual base. Can you tell me why? Yes, can you tell me why? Krutika, Niharika, anyone here in the class? Why this base is unusual? Because here you will see triphosphates. Normally in the DNA or in the RNA we see monophosphates, they are nucleoside monophosphates, but it is having triphosphate, so this is unusual. And enzyme guanyl transferase, right? And enzyme guanyl transferate will help in this feat. Okay. Okay, this is the enzyme which will help in adding this unusual base to the 5' end, right? So this is the capping. This is the capping. And one more thing, this 7-methyl guanosine triphosphate, this 5' end will also help in translation. You know why? Because it is having sites for binding to smaller subunit, to the smaller subunit of ribosome. Do you know that? So this cap is helpful in the translation. How? Because it will bind to the smaller subunit of the ribosome during translation, right? So it is having the site for binding to the smaller subunit of ribosome. So this is capping. Now when it comes to the tailing, tailing. Obviously tailing will be done at 3' end. Tailing means addition. Addition of what? Yes bache, tailing means addition of polyadenylate, addition of polyadenylate, or you can say that addition of adenyl at 3' end. This is the tailing. So now let's say here you have your mRNA. This is the 5' end, having that cap, having that 7-methyl guanosine triphosphate. Okay, so now to this 3' end bache, so many adenyl groups will be added. Do you know that? So many adenyl groups, so many adenylate residues will be added. Right bache, very good. So many adenylate residues will be added at 3' end. And do you know what? It will be done in template independent manner. This is important. It is an important question bache. That will be done in template independent manner. Okay, this is important MCQ. Okay, so which enzyme is going to do that? That is PAP. PAP means polyadenylate polymerase. Which enzyme it is, students? It is PAP, polyadenylate polymerase. Which enzyme it is? It is PAP, polyadenylate polymerase. So what is the meaning of adding the bases? 200 to 300 residues will be added to 3' end, right? But in template independent manner. What is the meaning of template independent manner? See, normally what we have seen, we have a DNA, complementary to it we are adding the bases. Complementary to it we are adding the bases. We have the DNA and complementary to it we are adding the bases. That is what we have seen so far, right? That is what we have seen so far. But here the point is, we do not need anything complementary to add adenyl. We have one enzyme that will just pick up that adenyl moieties and it will and this enzyme is going to add it here at the 3' end. That's how it is working. Fine, Niharika, that's how it is working. So this is the capping, this is the tailing, and you already know about splicing. So that will come under the RNA processing, and after RNA processing, your heterogeneous nuclear RNA will become mature RNA. Is that clear? Your heterogeneous nuclear RNA will become mature RNA. Is that clear? If it is clear, I want to see the energy in the chat section, guys. Be quick. Yes, be quick. If you understand it, be quick, because after that we are going to start the topic genetic code. Okay, after it, this is what we are going to start. We are going to start the genetic code. So tell me, people, if it is clear to you. Yes, so template independent manner word is important, unusual base is important, this entire process is important. And bache, this capping and tailing is going to protect the mRNA from the nucleases present in cytoplasm. Understood? This capping and tailing is going to protect the mRNA, right? This is going to protect the mRNA from nucleic acid digesting enzyme, that is your nucleases present in cytoplasm, right? So that is how it is going to protect. And splicing means removal of introns. So any doubt? Any doubt? So I hope this part is clear to you all, people, right? So what is a transcription unit? What is a gene? We have already covered. So gene is nothing, it's the functional unit of inheritance, another MCQ. Okay, right? So DNA sequence coding for tRNA and RNA is also define a gene. However, defining a cistron as a segment of DNA, coding for polypeptide, the structural gene could be said as monocistronic in eukaryotes or polycistronic in prokaryotes. So they have used the word mostly. So if we are using the word mostly, it means exceptions are also there. Let's not talk about that, right? Let's not talk about that. So what are eukaryotes? Are they monocistronic or are they polycistronic? Hanji, Kavi, Leharika, Neharika, Anjani, everyone in the class. Eukaryotes are monocistronic or polycistronic? They are monocistronic. They are monocistronic. Right? Sure? Very good. Very good. So coding, so in eukaryotes, monocistronic structural genes, they are interrupted right? You know that the genes in eukaryotes, they are split. So see, we have covered this concept as well. है ना? We have covered this concept as well, isn't it? So coding sequences or expressed sequences are defined as exons, right, which will give you the mature or processed RNA. They are interrupted by introns, so they are the intervening sequences which are not there in processed RNA. Okay, okay, right? So bache, this point is also important, that inheritance of a character is also affected by promoter and regulatory sequence of a structural gene. Promoter and regulatory sequence is there in that, na? In that particular transcription unit we have discussed. So sometimes the regulatory sequences are loosely defined as regulatory gene, even though these sequences do not code for any RNA or protein. So please do not miss such lines from NCERT. Okay, right? So they are simply saying that regulatory sequences are nothing, they are the regulatory genes, and these sequences do not code for any RNA or protein. Okay, so now the next thing, let's talk about the types of RNAs, and then let's talk about the genetic code. But before that, please have a look. See, everything is from NCERT. So this information is from NCERT. Important. Okay, right? So diagram is also important. Exon, intron, 7-methyl guanosine triphosphate, right? So see the spliceosome. This is the spliceosome. Fine. So spliceosome is a large RNA protein complex that catalyzes the removal of introns from nuclear pre-mRNA. Done? Yes, students, is it clear, or do you have any other doubt? Do you have any other doubt? So see, this is the NCERT. We have already covered everything. Okay, so if there is any issue, if you find anything difficult, you know, you can ask me the questions, right? You people can ask me the questions, right? Right. So see, what are they saying? That RNA polymerase binds to promoter, initiates transcription, it uses nucleoside triphosphate as substrate, and polymerize in a template dependent fashion. Means the formation of mRNA is template dependent. The DNA information will be used to add the bases, right? So it somehow also facilitate opening of helix, means your RNA polymerase. So only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA will fall off, it will get separated, right? And the RNA polymerase also, and this is the termination of transcription. So still, if there is any problem in the in understanding the language of NCERT, right, just let me know in the comment section, bache. I will help you. Okay, fine. Okay, so what is the importance of split genes? Tell me. This paragraph is all about RNA processing, so you can mark it in your NCERT, right? You all can mark it in your NCERT. What is the precursor of eukaryotic mRNA? Obviously, NEET aspirant, it is hnRNA, bache, right? So see the significance of split genes. The significance of such complexity is now beginning to be understood. The split gene arrangement represents probably an ancient feature of the genome, right? It is telling us about some primitive feature. So presence of intron is reminiscent of antiquity, and the process of splicing represents the dominance of RNA word. So basically this splicing is telling us about what? About the RNA word, that how important that RNA is for the formation of the protein. That's why it is important. If mRNA, it is not having the functional information, we will not get the protein, our system will not work. Okay, the system will not work. Clear, bache? So in recent times, the understanding of RNA and RNA dependent processes in the living system has assumed more importance. So somewhere it is related to that RNA word concept, right? So next topic that we need to start is genetic code. But as I said, before starting the genetic code, let's talk about the types of RNA. And yeah, we have already completed the process of transcription, so let's discuss that. Fine, let's discuss that. Okay, so are you ready? Tell me, are you ready? Yes, ma'am. Let's discuss it, and then I'll give you a break, and after the break we will start genetic code and the translation. Then again break. After that break, your HGP and your operin and your DNA fingerprinting. So we are going to finish it very quickly. Okay, okay, we are going to finish it very quickly. Done? Fine. So let's start with the ribosomal RNA. So it is the most stable type of RNA. Do you know that? Ribosomal RNA. Yes, we are going to talk about the different types of RNAs now. So do you know that your ribosomal RNA? Actually, this is how we divide. One is your genetic RNA, where your RNA is behaving like the genetic material. Another word is your non-genetic RNA, where RNA is playing other roles. So here we have the example of ribosomal RNA, we have the example of tRNA, we have the example of mRNA, right? We have the example of mRNA. So bache, when you talk about the ribosomal RNA, you know that it is the one that will make the ribosome, isn't it? Isn't it? It is making 80% of total RNA of cell. Do you know this thing? It is making 80% of total RNA of cell. Do you know that? Yes. Do you know that 80%? Right? So it is found in ribosome. You know that along with some protein, about 80 proteins. It was a question in NEET 2023. Right? How many proteins are there? 80 proteins are, near about 80 proteins are there in that ribosome. So it is found in ribosome and synthesized in nucleolus. Remember? Right? Remember? 5S ribosomal RNA. That is a kind of exception. It will not be synthesized in nucleolus. Okay, it will not be synthesized in nucleolus. Clear, bache? Clear, bache? So you know that when you talk about the prokaryotes, see, in the prokaryotes you have 30S subunit as the smaller one and you have 50S subunits at the larger one. So please do not add it. 50 plus 30 is 80. No. This S is telling us about the sedimentation. It is the Swedberg. Right, it is the Swedberg unit. It is a sedimentation coefficient. During cell fractionation, it tells us that whether that subunit is heavy or light and where it got settled. That's the point. Okay, so it's the Swedberg unit. So 50S is made up of 23S ribosomal RNA, 5S ribosomal RNA, and 34 proteins are there. And in the case of smaller subunit, in prokaryotes, 16S ribosomal RNA and 21 proteins. Right, bache? Right, bache? Now when you talk about the eukaryotes, small subunit is made up of 18S ribosomal RNA. It is 40S. Larger is made up of 28S, 5.8S, 5S. Clear? 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 So if you look at the proteins in eukaryotes, they are near about 83. So you should know about it. You should know about it. Fine. You should know about it. This information is important. Understood? This information is important. Clear, bache? Sure. So yes, it is the most stable form of RNA and most unstable is your mRNA. Most unstable is your mRNA. So just look at this mRNA. See, look at this mRNA, the most unstable form. I already told you it makes 1 to 5% of total RNA. Right? It is the messenger. It is having the information for making the protein. Right, bache? So just look at the structure. In prokaryotic mRNA or even if it is eukaryotic mRNA. Bache, do not forget about the UTRs. We will also talk about it in translation. We are also going to discuss about it, these UTRs in translation. Do you know what are UTRs? Do you know what are UTRs? UTRs means untranslated regions. Untranslated regions. UTRs. Like introns, they will also not give us the information for the formation of a protein. They are also not going to give us the information related to the protein formation. But still, they are the part of mRNA. They are the part of mature mRNA also. They will just help in the ribosome binding to the mRNA during the protein synthesis. So somewhere they are increasing the efficiency of that translation. right? They are increasing the efficiency of that translation. Clear? So when you look at the prokaryotic mRNA, you can see at 5' end, just before the start codon. You know na, start codon is going to be AUG, methionine. This is the start codon. Let me start the genetic code, you will understand it in a better way. Right? AUG, methionine, it is the start codon. So just before the start codon, you people will get untranslated region at 5' end, and just after the stop codon also you will get untranslated region at 3' end. And look at this mRNA, right? Because you know that prokaryotes are polycistronic, so see, 3 different proteins you are getting. Now come to the eukaryotic mRNA. This diagram is very beautiful, right? And this is some sequence. I'll talk about this sequence in translation. This is Shine-Dalgarno sequence, SD sequence. We will discuss about this sequence in translation. Okay, so now look at this eukaryotic mRNA at 5' end, 7-methyl guanosine cap, which will help it to bind to the ribosome. Then again, after this cap, you have untranslated region and then the start codon. And here, even after the stop codon, where is the stop codon? Here, right? You have 3' untranslated region. So these UTRs are present in both prokaryotes as well as eukaryotes. Prokaryotes polycistronic, eukaryotes monocistronic. So this is about the structure of mRNA. The structure of tRNA we will start just before the translation. So now let's talk about the genetic code. So do you need break or not? If it's a no, let's continue then. Do you need a break? Even I don't need, but still, you better take a break so that, right, you can quickly revise the transcription from NCERT, right? So break time is there, and break will resume at 7, it's 7 na, will resume at 7:20, fine. Will resume at 7:20. Then after finishing the genetic code and the translation, we will have another break. Fine. So please revise this topic for some time. Okay. So hi everyone, welcome back. Let's continue the session and let's start with the genetic code. Okay, so yes, for sure you're going to get question from genetic code and from Human Genome Project. Both the topics are equally important and that is what we need to discuss, genetic code. So tell me, are you excited? Yes? Are you excited? So let's finish the genetic code quickly. So tell me, what do you understand by this genetic code? Whatever you know, just let me know in the chat section, everyone. And yes, let me remind you this thing, guys. Please watch this video. 3 months, right? 3 months enough to crack NEET 2024 or not. You will get an idea that actually is it possible or not. Right, you will get a proper planning, proper strategy. So this is the video that you have to watch. And tomorrow, right, because today we will finish this chapter. Okay, today we will finish this chapter. So tomorrow at 11 AM there will be a special class where we are going to discuss practice the questions on the basis on molecular basis of inheritance. Last time some technical error was there, so that's why class ended, but tomorrow we will practice all the questions, all the last 10 years or 15 years PYQ from molecular basis of inheritance. So do remember, bache, and be the part of that class. The link is given in description box, and Ambika 10 is the coupon code that you have to use for joining that particular, right, for joining that particular class. Okay, and yes, do join this batch before it's too late. Right, the NEET endgame. Again, the link is there in the description box. Okay, so let's start the genetic code. Fine, let's start the genetic code. So now, as per the central dogma of information, I will repeat central dogma again and again. And even it is the favorite question of examiner. So DNA will give you mRNA, mRNA will give you protein. That is the central dogma of information. So first is transcription, and the second is translation. So bache, before starting the translation, we really need to know about the genetic code. Now we know that mRNA is going to make the protein. Bache, mRNA is what? mRNA is what? mRNA is a polynucleotide chain, isn't it? What is it? It is a polynucleotide chain. It is a nucleic acid, right? Right? And now on the other hand, when you talk about the protein, what is a protein? Protein is a polypeptide. Protein is a polymer of amino acid. Okay, it is a what? It is a polymer of amino acid. For amino acid, I'll be using the short form AA. Okay, it is what? It is the polymer of amino acid. Now when you look at this, right, one is a polynucleotide chain, another is a polymer of amino acid. Imagine from a polynucleotide chain we are getting a polypeptide, right? And that relationship is explained by this genetic code. So this genetic code explains this thing, that how a polymer of nucleotide forms a polymer of amino acid. Is that clear? Is that clear? Right, bache? So that relationship is actually explained by what? That relationship is actually explained by your genetic code. So if you have any doubt, please ask me, because genetic code needs to be very clear to you. Yes, bache, genetic code, it should be very clear to you, and trust me, it's a very simple topic as well. Okay, okay, right? So genetic code is what? It is the relationship in between the polypeptide chain and it is a relationship in between the polypeptide chain and the polynucleotide chain. That is what the genetic code is. Is that clear? And now write down, this is also important, the term genetic code was given by George Gamow. Who gave this term, bache? It was given by George Gamow. He was a physicist. Okay, and as per him, 4 bases, right? as per him 4 bases are going to give him, yes bache, 4 bases are going to give are going to form the 20 amino acids. Do you know the name of the 20 amino acids? Yes bache, do you know the name of that 20 amino acid? Anyone in the class? Do you know the name of that 20 amino acid? Quick. Quick. Anyone? The name of 20 amino acids? बचे alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenyl, alanine, right? Serine, threonine, tryptophan, tyrosine, and the valine. Okay, these are the 20 amino acids. Magic, right? We also call them as magic amino acids because they are going to make your proteins. It's not like that we have only 20 amino acids. There are more than 20, 200, 300 amino acids are there, but these are the protein making, the proteins that we need. These are that protein making amino acids, right? And I challenge you to remember the name of these 20 amino acids if you can. Again, I'm repeating this: alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, threonine, tyrosine, tryptophan, and you have the valine, right? Right? So remember the name of 20 amino acids. Maybe I have missed the one. I don't think so, but okay, right, bache? So basically that amino acids, they are the one that are going to make the protein. And how that amino acids are synthesized, that information is there in mRNA. So we have to pick up the 3 codons. That's what we know. But it was not as simple, right? Many scientists, they worked hard and then they explained this thing. As I said, the genetic code explains that relationship. The term was given by George Gamow, and George Gamow, he was the physicist, right? He pointed out the possibilities of 3-letter codes. Do you know that? Yes. He pointed out the possibility of 3-letter code. Okay, bache. So actually this genetic code, it was discovered by the scientist name, their contribution, everything is important. So it was discovered by Nirenberg, Mathai, these are the name of the scientists, and the Indian scientist, of course, Hargobind Khorana. So they discovered this, right? They discovered this genetic code, and the possibility of having 3 bases, right, to form that codon was given by George Gamow. So just look at it. Let's read it from NCERT. So during replication and transcription, we know nucleic acid will be copied to form another nucleic acid, which is again easy, right? So these processes are easy to conceptualize, right, because complementarity is there. But for translation, we have to transfer the genetic information from a polymer to nucleotide, polymer of nucleotide to polymer of amino acid. Of course, it's a major change, guys. Isn't it? It's a major change. Genetic information need to be transferred from a polymer of nucleotide to the polymer of amino acid, right, where there is no complementarity, right, bache, nor could be drawn theoretically. There is nothing, there is no common connection, right? So there are sufficient evidences which support the notion that change in nucleic acid, like if there is any slight change in genetic information, it will change the protein. So means there is a relationship in between, isn't it? There is a relationship in between. Clear, bache? Clear, bache? So if determining the biochemical nature of genetic material and structure was very exciting, the proposition and deciphering, right, deciphering of genetic code were most challenging. Clear, bache? So various scientists, they interacted together, they involved together, like physicists, organic chemists, biochemists, geneticists. Then it was George Gamow, a physicist. It's important. George Gamow, a physicist who argued that since there are only 4 bases, so if they have to code for 20 amino acid, the code should constitute a combination of bases. He was the one, right? He was the one who suggested that. Clear, bache? So he suggested the possibility that 3 nucleotides, the combination of 3 nucleotides can give you the 20 amino acids, right, bache? 20 amino acid. Now it's very simple. We have adenine, guanine, cytosine, uracil, because RNA is having this now. Now let's say if we consider two bases, combination of two bases. So see, first position can be filled by any of the four bases. Second position can also be filled by any of the four bases. So it means it will code for only 16 amino acid, right? But it is not solving our purpose, right? It is only coding for 16 amino acid. Again, it is not solving our purpose. Let's say if we take the combination of three bases, right? This first position, again, any out of any four bases, any base can come here. Out of that four bases, any base can come here. Adenine, guanine, cytosine, uracil, any one can come here also, here also. So total 64 codons will be there. Again, bache, it is helpful. Again, bache, it is helpful because if we are getting 64 combinations, 64 codons, right? No doubt, right? Definitely from these 64 codons, we will get our 20 amino acids, isn't it? There is a possibility that we can get our 20 amino acids. Right, bache? Right, bache? So basically, we have our codon is what? Right, codon is having three bases. It's a triplet base codon. Right, it's a triplet base codon. Three bases are making one codon and they are coding for one amino acid. Right, they are coding for what? They are coding for one amino acid. Clear, bache? Clear, bache? Yes. Yes or no? And you know that, let's talk about the chemical methods as well, right? So providing proof that codon was a triplet was a more daunting task, right? They mark the possibility that yes, it is possible, but they have to prove it, right? Right. So the chemical method, this is important, developed by Hargovind Khurana. Bache, in the final NEET paper, they can ask you the contribution of each and every scientist. So please pay attention. So the chemical method developed by Hargovind Khurana, it was instrumental in synthesizing RNA molecules with the defined combination of bases. So what he did, he developed the chemical method, right, bache, and because of that we synthesized the RNA molecules. So he picked up homopolymers, like let's say all the bases are same. You know that in that scenario, when three bases will be picked up, they will give the same amino acid. Like let's say if it is the copolymer, right, let's say if this is the combination, they picked up Cu, but we know how, right, it will be CuC, then UCU. Then again it will be CuC, UCU, CuC, UCU. So it will give us one amino acid, like amino acid 1, amino acid 2. Again, amino acid 1, amino acid 2. So that is what we are going to get. So that's how they synthesized different RNAs and they came to one conclusion. Then bache, Marshall Nirenberg, he formed a cell-free system. What he did, he formed a cell-free system. Like basically proteins are synthesized within the cytoplasm in a cell, but he formed a cell-free system. We can synthesize protein even outside a cell. We don't need a proper cell. Clear, bache? Clear, bache? And obviously it was important to decipher that code, that how that proteins are getting formed. Right, bache? And then bache, Severo-Ochoa enzyme, that is polynucleotide phosphorylase. It is important, right? This everything written here is important. Please mark it in your NCERT. So Severo-Ochoa enzyme, that is your polynucleotide phosphorylase, it was also helpful in polymerizing RNA with the defined sequences, but the beauty here is template independent manner. Right? So with the help of enzyme, that is your polynucleotide phosphorylase, the RNAs were synthesized. Right? And finally they made a checkerboard for it. Right? So in that checkerboard, bache, we will see 64 codons. Okay, what are we going to see? We will see 64 codons. Codons means, you know that one codon is triplet, means in each codon there will be 3 bases, right? What will be there? There will be 3 bases in each codon. Understood? There will be 3 bases in each codon. So bache, out of these 64 codons, it's very simple. Let's say if there are 20 amino acids, so 64 minus 20, it will be your 44. It will be your 44. So 44 minus 3 codons. 3 codons, they are the stop codons. So you are left with 41 codons. So what is the role of these 41 codons? So we need to understand everything, so that checkerboard will help us to understand that, right? Do you know how to make that checkerboard? Yes, do you know how to make that checkerboard? It's very easy. It's very easy. I'm not telling you to remember this checkerboard, but if you can, that will be helpful, because sometimes we used to get questions, right? Sometimes we used to get questions, right, bache, where, you know, yeah, sometimes we used to get questions where we discuss that thing. What we discuss? We get the questions where they ask you, they'll give you the codon, they'll ask you this codon is going to give you which amino acid. So if you are interested in scoring 360 out of 360 marks, so you cannot miss anything. Okay, one thing. Secondly, bache, the amino acids which are written in NCERT, right? Which are written in NCERT, जैसे serine, tyrosine, methionine, tryptophan, lysine, leucine, whatever is written, arginine. So the names which are given in NCERT, at least you should not miss them. You should know the codons that code that. Okay, I'll give you the trick. I'll provide you the trick. Tomorrow I'll be posting one more video for where we will be discussing the complete genetic code with the tricks. Okay, I'm going to post that video where we will be discussing the genetic code in detail with the help of tricks. But right now, please focus here. So let's make this checkerboard. It's very easy. How many bases do we have? How many bases do we have? 4, right? Right, we have 4 bases. So you'll take 4 columns. Right? It's very easy, trust me. So what's the first trick here? The first trick is you can arrest gangster. You can arrest gangster. Right? You can arrest gangster. So from this U, you will get U. From this can, you will get C. Arrest A, G, G. Right, so this is the sequence UCAG. Fine, this is the sequence UCAG. Here write down UCAG. Here also UCAG, and here also it will be UCAG. Right? That's how you have to write it first. Is it clear? That's how you have to write it first. UCAG, 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 UCAG. Thank you, bache. Thank you, Madhushri. Right? You have to keep the same sequence. Actually, this side, your codon is, right? Your codon is having 3 bases now. So this side is telling you about the first base. This side is telling us about the second base. Okay, this side. And this one is for the third base. This one is for the third base. And you know that, right? If you know it, very good. Otherwise, let me tell you, whenever we read the mRNA, right, while synthesizing the protein, we always read the mRNA in 5' to 3' direction. Right? We always read the mRNA in 5' to 3' direction. The ribosome will translocate in 5' to 3' direction. So as I said, here this side is having first base, this is having second, this is having third. So first base means it is telling us about the 5' end. Okay, last base, your third base, means it is telling us about the 3' end. So now what's the next thing? What's the next thing that we have to do? It is telling us that first position, first base means at first position you have U, at second position you have U, at first position U, second U, first U, second U, first U, second U. Now when you come to this side, right, randomly I'm writing it. First position U, second base should be A. First U, second A, first U, second A, first U, second A. Now what about the third base, ma'am? In this sequence you have to write it. UCAG. In this sequence you have to write it. So simple as that. That's how you can make this checkerboard. So first of all, draw this, right, toward this side, UCAG, UCAG, and here also UCAG. So basically this will tell you about the position of, this will tell you about the first base, this will tell you about the second base, this will tell you about the first base, the second base, and the last will be like this, in this order, UCAG. Okay, is it clear or not? Okay, let me tell you about this. Here, which one is your first base? Right, I'm talking about this side. Which one is your first base here? Kavi, which one is your first base here? Tell me, U, right? What about the second base, C? So UC, 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 right? And when it comes to the last base, it is going to be UC, AG. Simple, simple. That is how you can write down all the, right? That is how you can write down all the 64 codons. So there is no problem at all. It's actually very simple. Trust me, it is very, very, very simple. I know no one is going to ask you to draw it, right, bache? But yes, with this way, right, bache, you can easily remember that which codon is coding for, right, which codon is coding for which amino acid. So this particular table that I'm explaining here is for those who are targeting 360 out of 360 marks, right? Are you people targeting 360 out of 360 marks? Tell me, are you people targeting 360 out of 360 marks? Tell me. If it is so, just let me know in the chat section, quick. See, it's very easy. You can also try it after this lecture if you want to. It's something very simple. Done? Done? AU not UU, where? CA. It is AU na? Here first base is A, second is U. Here first base is U, second is also U. And here also first, okay. This one you are saying, very good, very good. First base is A, second is C. So ACC, ACA, very good, thank you. Is it clear? Is it clear? Yes, so CA, 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 UA, 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 and here AA, and here GA. So here also UG, CG, AG, and GG. Done? Done, done, done. So here see, in this column, second base will be U. Here, second base will be C. Third base will be A, G, like this. Okay? Third column. Bache, third column is fine. First, second, third column. Are you talking about this one? First base U, second is A. First base C, second is A. First base A, second is A. First base G, second is A. What is the mistake here? There is no mistake. Which one? Which one are you talking? Is it clear? Okay, so see this one, this is my favorite. Second column, the trick here is sar pro thar ala. Repeat everyone, sar pro thar ala. Sar pro thar ala means this one, all the four codons are coding for sar, means serine, right? Sar pro means pro, PRO, prolin, right? Prolin. Thar means here THR, threonine. We are talking about the threonine here. Sar pro thar ala, ALA means alanine, right? Alanine. So bache, these three letters, right? These three letters are also telling us about the short forms, and there is one letter code as well. Jaise ki your alanine, A. Arginine is denoted by R, right? Right, bache. This is what right, this is what I'm telling you. So basically when you talk about the amino acids, they are having these 3-letter code as well. This is SER, SER, serine, proline, threonine, alanine, SER, protharala, SER, protharala, SER, protharala. So all these 4 codons coding for serine, they are proline, threonine and alanine. These 2 are coding for PH. Yes, can you tell me what is it? PHE, phenylalanine, right bache, it is phenylalanine. Next 2 are coding for leucine, LUC means leucine, right? So leucine is having maximum, okay, leucine is having maximum codons. So these 2 LEU and these 4 also LEU, leucine, right? So I'll give you the trick but in another video. Fine. So just look at this, I hope now this table is clear to you all. First position, second position, third position. So that will be your 5' end, that will be your 3' end. So first base will be U here, U. And you know that last will be UCAG. So second base here is U and U. Look at this. First base is U, second is A, UA. First base is A, second is A, like this. Clear? Like this. So this is that checkerboard. So let me tell you the points which you need to focus, and then we will talk about the salient features, and then we will start the translation. So please pay attention here. Right? There are some points. If you can remember all the codons, it's very simple. You should know how to draw the table, and then you can make the trick. Like here, this is working for me. SER, pro, thar, ala. Right? SER, pro, thar, ala. Clear, bache? So this is the 3-letter code, but we have single letter code as well. Like I said, alanine, for alanine you can also write A, but for arginine it's not A, it's R, like this, right? So you have these codes, right, for all the amino acids. Now what you need to write down, first thing, your methionine, right, MET, this one, it codes for methionine, and this is your initiator codon. What is the meaning of initiator codon? Means it is the start codon. It is something which will start the protein synthesis. All the time, whenever there will be the protein synthesis, this is going to be the start codon. This is going to be the start codon. So it is having dual function. It is coding for methionine as well, and it is the start codon. And sometimes bache, your GUG codes for valine, right? So it can also act like a start codon. Not always, but yeah, it can also, right? It can also act like a start codon. Is that clear? Now another important point is that only methionine and tryptophan, please write down, only methionine and tryptophan are coded by, only they are coded by one codon, right? This information is important. You know that AUG is coding for methionine, no other codon will code for methionine, and same for the tryptophan. Can you see this? UGG, AUG or UGG, it is for tryptophan. So only they are coded by one codon. Rest all the amino acids are having more than one codon, right? Rest all the amino acids are having more than one codon. Understood? Maximum codons are of leucine, right? Total 6 codons are there for the leucine. Yes, total 6 codons are there for the leucine. Can you see that? 6 codons. So it is having maximum, right? It is having maximum. Understood? And then bache, stop codons, right? This is what you need to remember, that there are 3 stop codons. Please, this is important. There are 3 stop codons. They are also known as termination codons. They terminate the translation. They are also known as nonsense codon. Remember, nonsense codon. Do you know how to remember this? Chetu, Kavi, Leharika. So it's U. That's how we have to write it. U. I'm repeating this again. It is important. It is very important. It's U. Then AG. AG means AAG. AGA. AGA. AAG. AGA. AG. So this UAA, it codes for these three are what? They are the stop codons. Right? So another important question is related to their name. So UAA, it is your ochre, UAG is your umber, and UGA is your opal. You should know about these names as well. That's important. Understood? That's important. Understood? So these three stop codons, methionine, tryptophan, leucine is having maximum codons, so you people should know about it. Clear, bache? You people should know about it. Done? So again I'm repeating, whenever we read the mRNA, okay, whenever we read the mRNA, let me tell you, AUG is your initiator codon, na? Let's say, so what are you going to read? You will always read it in 5' to 3' direction. So we pick up first 3, next 3, next 3 and next 3. Means there is no overlapping. Do you know that? There is no overlapping. You cannot do it like this. AUG and then it will be UGU, then it will be UUU, or it will be GUU. No overlapping is there. We have to consider first 3 codons, then next 3, then next 3, then next 3, like this, right? Like this. So there is no overlapping, right? Your genetic code is commaless. Basically there is no punctuation at all, and obviously it is having universality, right? It is universally acceptable. Even if you are talking about the prokaryote or you are talking about the eukaryote, right? The UUU is going to give the phenylalanine only, right? So this is the meaning of being universal. Okay, so even if you are talking about the prokaryotes or the eukaryotes or anything, what are you going to see? You people are going to, right? What are you people going to see? You people are going to see that these codons are coding for the same amino acids everywhere. Okay, for same amino acids everywhere, right? There are few exceptions only. Fine. So the features are important, right? So first is the codon is triplet. 61 codons code for amino acid, and 3 codons, they do not code for any amino acid. They are the stop codons. So again in the NCERT, please write down the name of that stop codons, because it is important. Then bache, after that, see, the code is universal, nearly universal. From bacteria to human, UUU will code for phenylalanine. Some exception to this rule rules are there in the mitochondrial codons and in some protozoans. Okay, there are few exceptions, otherwise it will be accepted like this. Okay, so it will be read in a contiguous fashion. There are no punctuations, no comma, nothing at all, right? AUG is having dual function. It is the initiator codon as well as it codes for methionine, right? And these 3 are the stop codons. Is that clear? Now there are 2 very important properties that we need to discuss. One is degeneracy and one is non-ambiguous or unambiguous nature of genetic code. Second is non-ambiguous or unambiguous nature of genetic code. This is what we need to remember. Degeneracy and this. Understood? Degeneracy and this. Let's discuss that. These are 2 very important properties. Okay, so first let's start with the non-ambiguous nature of genetic code, right? Firstly, we should start with this non-ambiguous nature of genetic code. Clear, bache? Ambiguity means doubtful. What is the meaning of ambiguity? Ambiguity means when something is doubtful, right? But genetic code is having no ambiguity. It is not at all doubtful. So in the paper, sometimes what happens, because we are overexcited, we relate the words. Let's say they have given you the option genetic code is ambiguous in nature. So sometimes we do not pay attention that what they have written exactly, right? If it's like genetic code is non-ambiguous, right? Genetic code is non-ambiguous, so it is correct. But if it is written a genetic code is ambiguous, it is an incorrect statement, because genetic code is non or unambiguous. There is no ambiguity. Now what is the meaning of this line? See, it's very simple. As per this, that one particular codon will code for a particular amino acid. One codon will code for a particular amino acid in all conditions, right? Just to simplify this, I am explaining this, bache. What is the meaning of non-ambiguous nature? It means one particular codon will code for that specific amino acid only, right? One particular codon is going to code for the particular amino acid only, right? So one codon specifies only one amino acid, not any other. Just say for an example, right, GUG, in all the possible conditions, GUG will code for valine. It is not going to code for a methionine, right? GUG is coding for what? GUG is coding for valine. So in all the possible conditions, it will code for valine only. It's not like that today GUG is not in the mood and GUG will start coding for the methionine. No, it's not like that. GUG means valine only. Just AUG, GUG. There is a difference of only one base at 5' end. But that doesn't mean that GUG will be like, chalo theek hai, today let's try to code for methionine. No, in all the possible condition, it will be coding for the valine only. Your UU is not going to code for anything. It will code for phenylalanine only. It's like that. Few exceptions are there, but otherwise this is the thing, right? This is the thing. Now second point is degeneracy, right? So many times students, they confuse these two properties, the property of degeneracy and unambiguous nature, right? They confuse these two properties. It's very simple. Let me explain this to you, right? Literally it's very simple. Non-ambiguous nature is saying one particular codon is going to code for that amino acid in every possible condition, but degeneracy is saying that one amino acid can be coded by more than one codon. Bache, this is not contradictory to each other. It's very simple. Trust me, it's very simple, right? Here they are saying that one codon will code for a particular amino acid in all the conditions, and here they are saying that one amino acid can be coded by more than one codon. This is different, right? Just say for an example, we were talking about the leucine. Leucine is coded by 6 different codons, right? Leucine is coded by 6 different codons, isn't it? Yes, it is coded by 6 different codons. C, U, C, U, C. These are that 6 codons. Am I right? These are that 6 codons, right? Right. So leucine. So imagine 6 different codons, they are coding for same amino acid. That is degeneracy. And they are not breaking that, they are not creating any ambiguity. Okay, they are not creating any ambiguity. UUA in all possible situations will code for leucine. UUG in all possible situations will code for leucine. CUG in all possible conditions will code for leucine. This is non-ambiguity. So degeneracy is saying that one amino acid can be coded by more than one codon. That explains na. Jैसे we have 61 functional codons. ठीक है? We have 61 codons, right बच्चे? So 3 codons See, normally we have 64 codons. You already know that 3 codons are the stop codons. So we are left with 61. We are left with 61 codons. So here in these 61 codons, one will code for methionine, another will code for tryptophan. Okay? One is going to code for methionine, one is going to code for tryptophan. Again, you are left with 59 codons, and still you have 18 amino acids. So obviously it is obvious that one amino acids, they are getting coded by more than one codon. Just leucine is coded by 6, right? Leucine is coded by how many? By 6 bases, right? Isoleucine is coded by 3 codons. Leucine is coded by 6 codons. Isoleucine is coded by 3 codons. Valine is coded by 4 codons, right? Serine is coded by 4 codons. Are you getting it? That is degeneracy. So any confusion in non-ambiguity, non-ambiguous nature and degeneracy? Yes, bache. Do you have any confusion here in non-ambiguous nature and degeneracy? Anyone? Understood? So why this degeneracy is there? It is basically because of the third base, right? The third base is considered as the vowel base, bache, right? It is considered as the vowel base, right? So this degeneracy is due to third base, right? It is due to third base, vowel base. I hope you know about the vowel hypothesis. I'll use it in translation. Look at these 4 codons. First 2 bases are same, only the third base is different. Even here also, first 2 bases are same, only the third base is different. This third base is responsible for this degeneracy, right? We discuss it in vowel hypotheses. I don't know whether you know it or not. Vowel hypotheses. Do you know about this hypothesis, bache? Do you know about this hypothesis? Do you know about this hypothesis? Anyone? Yes? Do you know about this hypothesis? It is also propounded by Crick. It is propounded by Crick, bache. So bache, here in this hypothesis, see, right, in translation you will get it in a better way. Okay, so you know that in tRNA, after this we will be discussing the structure of tRNA, but just giving you an idea of it, right? If you know a little bit about tRNA, you know na, here it is going to have anticodon, here it is going to have anticodon. So in this anticodon, right, the first 2 bases, okay, right, the first 2 bases, they will always follow the, right now I am not specifying 5' or 3', in general I am explaining this, that first 2 bases will follow Watson and Crick pairing, right? First 2 bases, they will always follow Watson and Crick pairing. What is the meaning of Watson and Crick pairing? Right, that adenine will always pair with uracil, cytosine will always pair with guanine, right? This pairing, they will follow a Watson and Crick pairing. But when it comes to the third base, the vowel base, right, it will show it can even pair with the unusual bases. Okay, the third base can also pair with the unusual bases, right? So third nucleotide of the codon is going to do that wobbling. Okay, bache, it is going to do that wobbling. Right, bache? Right, bache? For an example, the anticodon AAG, right? Let's say we have this anticodon AAG, right? It can pair with both. Do you know that? It can pair with both, right? This anticodon can recognize both the codons. It can recognize both the codons. Can you see that? Can you see that? It can recognize both the codons. AA is following the Watson and Crick pairing, uracil, but this guanine, it can also pair with uracil, it can also pair with cytosine. Can you see that? Yes, bache, can you see that? So this is actually the wobbling. It is because of the third base. It is because of the third nucleotide or the third base. So it is showing unusual base pairing. It can also pair with cytosine, which is normal, but it can also pair with uracil, which is not normal. So right, so that is the reason, right? During protein synthesis, we will see we have 20 amino acid, 20 tRNA, but we have more than 20 codon. right? So this explains that. Fine, 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 fine. So vowel hypothesis clear, vowel hypothesis clear. Sure, bache? Are you sure? So now come to this part, that is your mutations in genetic code. I hope you remember this topic from Principles of Inheritance and Variation, right? I hope you remember this topic from Principles of Inheritance and Variation. Do you remember this part? Yes, from Principles of Inheritance and Variation. Sickle cell anemia, right? Sickle cell anemia, which is a qualitative disorder, right? Which is a qualitative disorder. So you know that in hemoglobin, yes bache, in hemoglobin in beta chain of hemoglobin, basically at 6th position, glutamic acid, right? At 6th position, glutamic acid will be replaced by valine, right? Glutamic acid will be replaced by valine. If you remember, GAG will become GUG. Do you remember that? GAG will become GUG, right? So this is the example of point mutation, right? At the place of A, U will come and it will change the codon, right? It will change the entire codon, the amino acid coded by that codon. So instead of glutamic acid, we'll be getting valine. Instead of glutamic acid, we will be getting valine. And you know that because of that, RBC will become sickle shaped, right? RBC will become sickle shaped, right? You can see this image also. Normally, glutamic acid is there at 6th position and here valine is there. Are you getting it? So this is the point mutation. What is it? Because we understand genetic code now, you understand genetic code now you can relate. You can relate. This is the mutation related to that genetic code. There was a change in that genetic code which changed the amino acid, which itself changed that RBC structure. Okay, which itself changed that RBC structure. So this is one of the examples. And moreover, right, this is point mutation, only one base got changed. If you remember frame shift mutation that we have discussed in the Principles of Inheritance and Variation, frame shift mutations, right? Reading frame will change. Like if 1 or 2 bases will be added, then they will change the reading frame. If 1 or 2 bases will be added or deleted, they can change the reading frame, right? If 3 will be added, comparatively the effect will be less, right? So in tomorrow at 11 a.m. we have a class now on Unacademy platform. You can see the link here in the description box as well, right? You all can see the link here in the description box as well. So we have a class tomorrow at 11 a.m. the special class. There we will practice such questions related to this, okay, related to this. So this is important, please pay attention, okay? This is important. So see, it is given in NCERT. You should know about these 3 letters code: valine, histidine, leucine, threonine, proline, glutamic acid, valine, histidine, leucine, threonine, proline, valine and glutamic acid. That's what you need to remember. And yeah, question can also come from this. They can give you these 2 polypeptide chain. They can also ask you which one is for HBA, which one is for HBS. Plus they can also ask you to put the amino acids there, right? So be prepared. That's why I added this image, right? So be prepared. Clear? So next type of tRNA, next type of RNA that we need to discuss is your tRNA, because now we have to start the translation. So we should know about the structure of tRNA first. Done? Then we will be talking about the translation, bache. After translation, the lac operans, HGP, finally DNA fingerprinting. Okay? So we just have 4 main topics, 4 topics only. Okay? 4 topics only. Okay? So are you ready to finish it today? And I think we can finish it till 11. We can finish it till 11 if you people support, or maximum by 12. Maximum by 12. Are you ready? Tell me, are you ready? Sure? Sure. So tell me, what do you know about tRNA? Quick. Tell me, what do you know about tRNA? What do you know about tRNA? quick. Yes, bache, when you talk about the tRNA, you are right, it is known as the transfer RNA, right? It helps in translation. It is also known as adapter RNA, right? It is also known as sRNA. It is not SN, it is S, means soluble RNA, right? Soluble RNA. And it makes near about 15%, right? It makes near about 15% of total RNA of cell. It makes near about 15% of total RNA of cell. Do you know that? Yes, bache, do you know that? So many questions used to come from this topic, okay? Yes. Okay, so here in this tRNA, it is playing role in translation. And why do we call it as adapter? Why do we call it as adapter? Because, bache, when you talk about this tRNA, na, it will help us to connect that mRNA. Like basically this tRNA will join to mRNA. It will join to an enzyme. It will join to amino acid as well. Okay, and it will also join to the ribosome. So it is just like an adapter, no? Adapter connecting the things. Okay, so it will also join to the mRNA, it will also join to the enzyme, it will also join to the amino acid, it will also join to the ribosome. Okay, ribosome. Now when you look at the tRNA structure, there are 2 points that you need to remember, right? It is having a 2D structure, means two-dimensional structure, and that structure looks like the clover leaf, right? Clover leaf like. And it is also having a 3-dimensional structure, but that 3-dimensional structure looks like the inverted L. That looks like inverted L. It is inverted L shaped. It is clover leaf like. See, look at this. This one is your 2-dimensional structure, right? This one is your 2-dimensional structure, right? It was presented by Holle. And this one is your 3-dimensional structure. It is basically the inverted L. So it is also a previous year question, let me tell you. It is also a previous year question. Here I don't remember the scientist name. Okay, so if you look at the structure here, especially the 2-dimensional structure, people, especially the 2-dimensional structure, can you see the structure is kind of folded? Yes, can you see that structure is kind of folded? And because of that folding, thank you Samriya. Right, thank you Samriya. Because of this folding, bache, because of Indu, because of this folding, right, some bases of the tRNA, they come closer, right? Because tRNA is like this, you know, normally RNA is single stranded. Because now there is a folding, and because of that folding, what is happening, people? What is happening? That some complementary bases, they come in front of each other. Right, they come in front of each other. So if they are coming in front of each other, obviously if they are complementary, they will form the hydrogen bonds, right? They are going to form the hydrogen bonds. So when they form such hydrogen bonds, they form such structures, these arms, right? When you look at this tRNA, na, when you draw this structure, this is arm, and when you draw this rounded structure, it is loop. Here in this loop, there is no hydrogen bonding, right? Here you have the bases, but this is loop, and this structure is arm, where two bases which are complementary to each other, they come closer and they form the hydrogen bond, right? Right. So now let's discuss the 2-dimensional structure of this tRNA in detail. Okay, let's discuss that. First of all, when you look at the structure, bache, okay, let me draw it in this way. I'm not writing the bases here. I will write the important things for you people only. Okay. This is your tRNA. What is it? It is your tRNA. So it is 5' end. It is your 3' end. Okay. Now you know that 3' end is always going to have free OH. Understand the tRNA structure. If you really want to understand translation, you should know the tRNA structure. Okay, if you really want to understand the translation. So please have a look. 5' end, 3' end. 3' end is having free OH group, right? 3' end is having free OH group. Yes or no? Yes or no? Right? This is what we already know. Now here, what is the speciality? I always told you that we read the codon in 5' to 3' direction. So when you see this, so here at 3' end, definitely you will get these bases CCA. Some people, they call it as ACC also. Always at 3' end. This is what you people are going to observe. See, this is the 3' end. You're always going to observe this thing, CCA. Right? You must be thinking that, ma'am, why can't you call it ACC? It's very simple. It's your choice. How do you want to remember? Right? It's your choice. Right? But the point here is, because we always read the codon in 5' to 3' direction, that is why I'm calling it as CCA. That's why I'm being very specific here. It's CCA. Fine, CCA. Right? Now when you talk about this particular arm here, it is amino acid acceptor arm. Which arm it is? It is amino acid acceptor arm. I told you your tRNA, right? I told you your tRNA is an adapter. It will join to all. So this is amino acid acceptor arm. The amino acid will join here. Now when you talk about this side, this loop is DHU loop, right? DHU loop, right? This is DHU arm. Okay, this one is T-Psi-C loop, or the arm here is T-Psi-C arm. This one is your variable arm. This one is your anticodon arm. Or this one, you know that this is anticodon loop. Anticodon loop. So just look at it very carefully. See, this is the amino acid acceptor arm. Means your amino acid is going to come here. It will form the bond with this OH. It will join here. First point. Opposite to it here, anticodon arm. Here it will be having the anticodon. So if it is having the anticodon, then obviously it will be joining with what? With the mRNA. Because the code is present on mRNA. Isn't it? Because the code is present on mRNA, and here this side will have the anticodon. So tRNA will check it, will bring the amino acid from the cytoplasm, and then finally the bases, the amino acids will be added in polypeptide chain. This is what you need to remember. Okay, so the function of this arm, these two arms clear. Now bache, variable arm. Right, but the function exactly of this variable arm is not known, but on the basis of this variable arm, we can divide tRNA into two different classes, class 1, class 2, right? On the basis of the number of nucleotides here in this tRNA arm, in this variable arm, we can divide tRNA into two different classes. Understood? Now DHU. The first question can come from this side is that what is the meaning of DHU? DHU means bache dihydrouridine. It's dihydrouridine. Like we have adenine, guanine, uridine, adenine, guanine, uracil, cytosine. So this dihydrouridine is also an unusual base. It is also an unusual base, and this side is having this. So we call it as dihydrouridine, right? This dihydrouridine is an unusual base. Now what's the point here, Chetu bache? It's very simple. It's very simple. This is my way to remember this. D for down. D for down. Down. When you are down, you need energy. You need energy. When you feel down, you need energy. When you feel down, you need energy. And for that energy, you need enzyme. Right? Who's going to provide you the energy here? The enzyme. Right? Basically, when you are down, you need charging. This is generally we discuss, right? Either you can pick up the word energy or you can pick up the word charging, whatever you want to. This is my way to remember that. So when you are down, you need charging, and for that charging you need, for tRNA's charging, we need the enzyme. So actually this DHU loop, it binds to enzyme. It binds to enzyme. So yes, you have to remember this. Right, it binds to what? It binds to enzyme. What is the name of that enzyme? That enzyme is amino acyl tRNA synthetase. What is the name of that enzyme? Amino acyl tRNA synthetase. Students, what is the name of that enzyme? Amino acyl tRNA synthetase. Now come to this T-Cy-C loop. T-Cy-C. The Cy means pseudo-uridine. The Cy means pseudo-uridine. Again, it is also an unusual base. It is also an unusual base. Now when this side is joining with mRNA, this side is binding to the enzyme, right, and this side with the amino acid, obviously this loop, this side will attach itself to the ribosome. It is going to attach itself to the ribosome. So that is what you need to remember. Again, clear bache? That is what you need to remember. Understood? Sure? Any doubt? Right? So ribosome mein, it is going to attach to the larger subunit of ribosome. Let's be more specific. It will attach to the larger subunit of ribosome. Fine? This T-Cy-C loop. So any doubt here in tRNA structure? It is very important. It is very important. Yes, you can again see this. Let's have a quick revision. Let's have a quick revision. D loop, D arm, D for down, we need charging. So the enzyme, right? Which enzyme it is? Amino acyl tRNA synthetase, right? This is amino acyl tRNA synthetase. This is the name of the enzyme, students. Clear bache? So T loop is going to join with the larger subunit of ribosome. Fine, larger subunit of ribosome. Is that clear? It is going to join with what? With the larger subunit of ribosome. Then this is the anticodone arm. Here it is having the anticodone, and here 3' end it is having CCA, and 3' end is having the OH group. Understood? And 5' end usually having this guanine. It is usually having this guanine G. Clear? Clear? Any doubt? Yes, if you still have doubt, you are free to ask. You are free to ask. Now look at this point. mRNA anticodone loop. You have 34, 35, 36, right? See, tRNA, and this is inverted L structure, right, bache? Okay. Now see your tRNA, 5' to 3'. Now when you are talking about your mRNA, 5' to 3'. So if tRNA, if it is having the anticodone, right, if it is having the anticodone, I hope you understand that much, that the base at 5' end will not join with the base at 5' end, or the base at 3' end will not join with the 3' end. Obviously the base present at 3' end will join the mRNA at 5' end, and this like this, and this like this. Is it clear or not? Is it clear or not? You know that your mRNA is, we have to study it in 5' to 3' direction. And here you have your tRNA. So tRNA, the base which is present at 5' end, the tRNA base which is present at 5' end, it will join to the base present at 3' end of mRNA. And here in tRNA, the base which is present at 3' end, it will join the base of the 5' end of the tRNA. Please observe this picture. I hope it will be clear to you people. Right, that why are they pairing this side with this side, this side with this side, and the middle one will stay same. Any doubt? Yes, any doubt? Sure, the structure of tRNA clear? The structure of tRNA clear? Sure? Are you sure? Done? So you know about the tRNA structure, you know about the mRNA structure. For your quick revision, let me remind you about the ribosome, right? So you know that in prokaryotes, that is the scenario. In eukaryotes, that is the scenario. Isn't it? Isn't it? So before understanding the translation, you should know these structures first. Right? So you know in eukaryotes, cytoplasm is having 80S ribosome, and mitochondria and chloroplast, they still have 70s. Here in prokaryotes, you are having 50s, 30s, right? This is the ribosome that you have in the prokaryotes. Hope you remember this. So you know that these two subunits will only join at the time of protein synthesis and only when magnesium ion is available. Do you remember that? Only when magnesium ion is available. So actually, right, see, when it is the prokaryotic mRNA, right, so to the prokaryotic mRNA, firstly smaller subunit will join, here also in the eukaryotes also, right, and then you know that further the processes will be done. We will discuss that in detail. So I would like to mention one thing here, that when you talk about the larger subunits, you are going to see 3 sites in the larger subunit. Do you know that? Right, when you talk about the larger subunit of ribosome, you will see 3 sites there, and the trick for that sites is E, P and A. It is E, P and A. The larger subunit is having what? Larger subunit is having 3 sites, that is E, P and A. E means exit site, we'll discuss their function. P means peptidal site. A means amino acyl site. These are the 3 sites which are present in larger subunit, and somewhere here there is one groove as well. In larger subunit, somewhere here there is one groove as well, and from this groove the polypeptide that has formed will come out. Okay, the polypeptide that has formed will come out. So up to this part all clear? Yes, up to this part all clear? So ribosomes, they will only join when there is the requirement of protein synthesis, right? Your larger subunit is having EPA, that is exit, peptidal side and the amino acyl side. There is a groove from where the polypeptide will come out. Clear, bache? And you know the tRNA structure as well. So if you have any doubt, do let me know. If you people have any doubt, do let me know. Trust me, today we are going to learn translation in such an easy way that you are going to love this chapter. Trust me on that. Okay, you people are going to love this chapter. You people are going to love this topic, and this lecture is more than sufficient. After watching this lecture, please revise the topic from NCERT and solve the PYQs. That's it, that's all. There is no need to refer to any other book. Okay, just go for the NCERT after it. Fine? So any doubt? Okay. First of all, I'm telling you the basics individually so that you do not mix up the things. That's what you know, 5' end, 3' end. 3' end is having OH, hydroxyl group, we know about that. Now let's talk about the basic structure of an amino acid. Right, let's talk about what? Let's talk about the basic structure of an amino acid. Do you know, bache, the basic structure of an amino acid? People, please let me know in the chat section. Guys, I want to see the energy in the chat section. Yes, I really want to see some energy in the chat section. Quickly tell me. Tell me quickly. Just tell me quickly. Yes, first of all, I really want to see some energy in the chat section, and then you have to tell me the basic structure of amino acid. Quick, you really want to finish this chapter? You really want to understand this chapter? Right, so when you talk about the amino acid, this is the basic structure of amino acid. This is the basic structure of amino acid. Yup. Is it the basic structure of amino acid? Definitely it is. Okay, so you know that there will be the change in this alkyl group, and it will decide that which amino acid it will be. Now, bache, here, when you look at this COH group now, that's important, please pay attention. When you look at the C double bond OOH group, you know that in amino acid you have the amino group and you have this carboxylic group, right? You have this carboxylic group, the acidic one. So actually now, when you talk about that amino acids now, so COH of amino acid will form the bond here, right? I told you that in the tRNA, this side is the amino acid acceptor arm. That's what I just said, that this side is going to be the amino acid acceptor arm. Yes, students, that is what I just said. Yes or no? That is what I just said, that it is the amino acid acceptor arm. It is the amino acid acceptor arm. It is the one which will accept the amino acid. Actually, whenever the tRNA will be charged, amino acid will be charged, this 3-OH group, with that 3-OH group, this amino acid is going to form the bond, people, right? It is going to form the bond here. That's what I'm telling you. Now, what is my point? Actually, from that amino acid, it is the COH, it is the carboxylic group which will form the bond here with this 3-OH. It is the amino acyl bond. Clear, bache? Right? So that's why it is the amino acyl bond that will form here. Is that clear? This is something very important. This is something very important, that the COOH of your amino acid will form the bond with the 3-OH of the tRNA. That's how this tRNA will pick up this amino acid. This point clear? So I am going to use a lot of slides here. Individually I will explain each and every step, so please pay attention. So the very first thing, whenever you talk about the translation, even if it is a prokaryote or the eukaryote, we know one thing, right? We know one thing. Even if it is a prokaryote or it is a eukaryote, we know one thing. And what is that one thing, Vardhan? What is that one thing? That the translation will take place in the cytoplasm. It will take place in the cytoplasm, right? Doesn't matter which cell it is, it will be there in the cytoplasm. For the translation, we will be needing, of course, the ribosome, the mRNA, the tRNA, the amino acids, magnesium ion, very important, right? And even some initiation factors, the factors that will help in the starting of the translation. Some elongation factors are also there, right? Some elongation factors are also there. Some release factors are also there. Okay, students, some release factors are also there. GTP, energy currency, basically it is also required, right? It is also required. This is what we need. So we are going to get it in the cytoplasm, right? We are going to get it in the cytoplasm. Clear, bache? Clear, bache? Please pay attention, right? You all are this all is available in the cytoplasm. So I told you this thing, and I think I repeated this thing so many times, right? I repeated this thing so many times that there are 20 amino acids. There are 20 amino acids. Actually, bache, now there are 22. We consider 22 amino acids, not given in NCERT, so we do not discuss. Two extra amino acids are there. Just say your 21st is selenocysteine, 21st is selenocysteine, and 22nd is pyrrolysine, right? 22nd is pyrrolysine. So selenocysteine and pyrrolysine, this is the 21st amino acid and 22nd amino acid. You should know about it. And if you are asking about their codon, you will be amazed to know, people, you will be amazed to know that in stop codon codes for these amino acids. In the stop codon, if there is, there are some elements present. I will not discuss that in depth. There are some elements present in that stop codons, right? If that particular element is there, that will code for selenocysteine. If that particular element is there, that will code for your pyrrolysine. So in that stop codons, right, they have some elements. If that elements are there, they can also form these amino acids. This is the kind of extra gyan that I am giving you right now, right? This is important. You can take the screenshot as well. Now, I'm telling you that we have to go as per our syllabus. So I told you we have 20 amino acids. How many amino acids do we have? We have 20 amino acids. So 20 amino acids means there will be 20 different tRNAs in the, right? There will be 20 different tRNAs, right, available in cytoplasm. Do you know that? We have 20 amino acids, right? Because every time for translation, a particular tRNA will pick up a particular amino acid. So we have 20 amino acid, we have 20 tRNAs. We have 20 amino acids, Alina, we have 20 tRNAs. Clear? Clear? Basically it is specific. If you are talking about the methionine will be having its own tRNA. If you are talking about phenylalanine, it will be having its own tRNA. If you are talking about cysteine will be having its own tRNA. If you are talking about proline will be having its own tRNA. So it is like that. Clear, bache? It is like that. Clear, bache? Right? So 20 amino acids, 20 tRNAs are there, and yes, there will be 20 different enzymes. Which enzyme? Amino acyl tRNA synthetase. So there will be 20 different enzymes. That will be your amino acyl tRNA synthetase, right? There will be 20 different enzymes. That will be your 20 different amino acyl tRNA synthetase. Understood? Understood? There will be 20 different amino acyl tRNA synthetases that are present. Is that clear? Is that clear? AA means amino acid. It's simple. Okay. Okay. Done. So now the first step of this translation. It is also a previous year question, right? They asked, they provided you 4 options, obviously, and out of that 4 options, this is what they asked, right? Out of that 4 options, that is what they asked. They even asked that which step is the first step. Okay, which step is the first step? So the first step is going to be the charging of amino acid, right? The first step is going to be the charging of amino acid. That is the first step. Okay. Okay, so again, you know that, right? You know that your initiator codon, the start codon, right? Which codon is the start codon? Initiator codon. The start codon is methionine. Yes, bache. It is methionine. है ना? Methionine coded by AUG. So let me tell you, in prokaryotes that methionine is formilated. Okay, in prokaryotes that methionine is formilated. That is actually known as formyl methionine. Right? 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 So start codon is always what? Start codon is always what? Start codon is always AUG, methionine. Right? In the case of prokaryotes, it will be formyl methionine. Formyl group is added to that methionine. So here comes one question, Elena, that it means, ma'am, all the proteins are having same first codon, है ना? है ना? If initiator codon is always the AUG, so that is what we can say, Shobhika. That is what, Shobhika, we can say that all the amino acids are going to have same first codon, right? Actually, when translation starts, it always starts with the help of that AUG, with the help of that start codon. But later on, in different type of proteins, you know, there are different mechanisms. Okay, so sometimes these amino acids, they get removed. It's not like that protein once formed, it will be like that only. No, it will be changed into another forms. So in that case, some amino acids will be removed as well. It's like that. Okay, so this part clear. So basically, first amino acid is my AUG. So obviously there will be a specific tRNA for that AUG. There will be a specific tRNA for that AUG. And for that AUG, there will be that specific amino acyl tRNA synthetase. Am I right or wrong? There will be the specific amino acyl tRNA synthetase. Yup. Isn't it? Isn't it? What I just said, Krutika, I'm just telling you that, right? I'm just telling you this thing, right? I am saying that you know that the first codon is going to be AUG. So it will be having its individual tRNA. It will be having its individual amino acyl tRNA synthetase. So, and yes, please make sure it is synthetase, not synthase. If you will write synthase here, that will be incorrect. Okay, if you are going to write what? Synthase here, that is going to be incorrect. It should be synthetase, right? Synthetase it is. Okay, so now the first step is the charging of amino acid. Ultimately, the process is, जैसे your enzyme ना, your enzyme is like a matchmaker. your enzyme, ultimately the role of this enzyme here is to put that tRNA on that, to put that amino acid on that tRNA. Basically, Alina, the role of that enzyme is to bring this tRNA and amino acid closer, right? This is what that enzyme is going to do. So the very first step that now we are going to discuss is charging of amino acid. Second is charging of tRNA. For both the steps, you will use only one amino acid tRNA synthetase, right? What do we have? What do we have here? Yes, what do we have here? We have two amino acids, right? What do we have here? We have two amino acids, right? We have two steps. First is charging of tRNA, second is first is charging of amino acid, second is charging of tRNA. For both the steps, you need a common amino acid tRNA synthetase. For both the steps, you need common amino acid tRNA synthetase. So as I said, first step is what? Charging of amino acid. So basically amino acid is going to react with the ATP, right bache? What is going to happen here? Your amino acid is going to react with ATP, obviously in the presence of enzyme. Obviously in the presence of enzyme amino acyl tRNA synthetase. Okay, and after reacting here, what are we going to get? We will get amino acid AMP enzyme complex, and that AMP means adenosine monophosphate, and obviously two phosphates will be released. So if I have to explain you this, it's like that. Let's say this is your amino acid, this is your ATP, and here you have your, let's say this is the let's say this is the enzyme, right? Let's say this is the enzyme. So in the first step, in the charging part, what is going to happen? To this enzyme, this amino acid will join, right? And obviously because this ATP, from this ATP two phosphates they are released, so AMP is helping there. So basically that AMP is also attached to this amino acid. That's the scenario, if you can understand. Yes, if you can understand. So what is happening here in the first step? What is the first step, Mani? It is the charging of amino acid, or you can also call it as amino acylation of amino acid, amino acylation or charging. Basically write it down as charging of amino acid. So amino acid, ATP, then amino acyl tRNA synthetase, then amino acid AMP enzyme complex, then comes the then comes this PP, right? Then what do you have? You have PP. So what's the point here, bache? This amino acid with ATP, here you have amino acyl tRNA synthetase, so they will form a complex. What kind of complex? Look at this enzyme. Yes, look at this enzyme. Can you see this enzyme? Can you see this enzyme? To this enzyme, this amino acid will get attached, and to this amino acid AMP is attached. So obviously this is a complex, no? Enzyme ATP, enzyme amino acid AMP complex, and two phosphates are released. First of all, please look at this. Please look at this. Is it clear or not? So a specific amino acyl tRNA synthetase is going to catalyze that reaction, okay? Right, so as I said, for each amino acid you have specific amino acyl tRNA synthetase. This is the first thing, okay? This is the first thing. Now the second point is charging of tRNA, and this is also known as amino acylation of tRNA. You can also call it as charging of tRNA. You can also call it as amino acylation of tRNA. If you know why am I calling it as amino acylation, do you remember now? 3' and 3' OH of tRNA will form bond with the carboxylic group of amino acid. 3' OH of tRNA will form bond with the carboxylic group of amino acid. Now here, what do you have? You already have that complex. Right, this is what we have, no? You already have that complex, right? Let me write it here. You already have that complex now. Yes, bache, this is what we have. Yes or no? This is what we have. So now that specific tRNA will come. That particular tRNA will come because we are just starting this process. So obviously here you can assume that it is for the methionine. But for all the steps, that is how it is going to proceed. So now what is going to happen? Here you have amino, my bad. Amino acyl tRNA, and the AMP will be released and the enzyme will be released, right? Look at this, please look at this. It is amino acyl tRNA, then AMP, then enzyme, right? Let me show you this part, bache. Let me show you this part. Okay, let me try to. So you know this, you understand this. This is amino acid, AMP, enzyme complex. Now you know your tRNA as well. You know your tRNA as well. So now what is going to happen ultimately, because of this enzyme's main function is to bring this amino acid to this tRNA. Enzyme is just like a matchmaker. Enzyme is just like, array amino acid, why are you getting worried? I'm here to fix you with the tRNA, right? And then after that, this enzyme will set itself free, okay, and even the AMP will be released. This is the meaning of charging of tRNA. That's why, Niharika, I am saying that we need the same enzyme. For the charging of amino acid, for the charging of tRNA, same enzyme is required, and it is going to be particular because it is the first step. I know you all know that initiator codon is methionine. So obviously we are doing all this for methionine. So every time when amino acids will be added, this step will be repeated with that particular amino acid. Right? So charging of amino acid, then comes the charging of tRNA. So here you can see when there is a charging of tRNA, ultimately this enzyme is going to put, right, this AA to this tRNA. How come? Because this tRNA will join with this enzyme. This enzyme will bring tRNA and amino acid closer, right? The COH of the amino acid will form the bond here with this 3-OH, and that is how the charging of tRNA will be there. So this is amino acyl tRNA complex, or simply you can write down amino acyl tRNA. Amino acyl tRNA means charged tRNA. Still, if there is any doubt, please feel free to ask. Please feel free to ask. Yes, please feel free to ask if there is any doubt. Now this complex will go towards the ribosome, right? This complex will go to the ribosome for the yes, bache, for what? For the translation. So now in the translation, the story is going to be same. But here in translation, the very first thing need to be done is the charging of amino acid and charging of tRNA. So first thing is the initiation. The first thing is what? It is the initiation. Right? The first thing is what? Initiation. Clear, bache? Right? So here you know what is going to happen in initiation. You can also write down. Bache, you know, when you talk about the prokaryotes, you know you have the 5' amino acid. This is the mRNA. This is what we know in the case of prokaryotes. Let's say here you have the start codon. You have the AUG. And you know that here it is going to be UTR. I think you all were there with me from the starting of the lecture, right? Right? So you know that when you people talk about the mRNA, wait, let me show you the picture. Picture. This one, I'm saying. So you know that here you have the start codon. Right, here you have the start codon, right, bache? Start codon. So just upstream to this start codon, here you have this untranslated region. I hope you remember this. Yes, I hope you remember this. So same way, right, let's discuss it. So when you are talking about the prokaryotes, 5' end, 3' end, here you have your AUG, the start codon, right? Here you have that UTR, untranslated region. So somewhere here in this mRNA, na, just before the start codon, here you will see a sequence. You know what we used to call it? It is known as a Shine-Dalgarno sequence. Shine-Dalgarno sequence, right? Obviously it is having that basis, no, it is Shine-Dalgarno sequence, SD sequence in short, Shine-Dalgarno sequence. Fine, Shine-Dalgarno sequence in the mRNA, right? Right? Where is it present? In the mRNA. Which sequence it is, bache? Shine-Dalgarno sequence. Now this sequence will be recognized by your 30S, you know na, 30S ribosome. We are talking about the prokaryotes here, right? We are talking about what? We are talking about the prokaryotes here. In prokaryotes, you know that smaller subunit is the 30S subunit, and 30S subunit is made up of which RNA? Yes, people, do let me know in the chat section. Niharika, Leharika, Mani, Alina, yes, Sabha, tell me, this 30S subunit is made up of which RNA? This 30S subunit, it is made up of which RNA? Tell me quickly. It is made up of which RNA? Excellent. It is made up of 16S ribosomal RNA. It is made up of 16S ribosomal RNA. So here in this smaller subunit, here in the 16S ribosomal RNA, it is having the sequence anti-Shine-Dalgarno sequence, means it can recognize this Shine-Dalgarno sequence. So why am I telling you this? I'm just telling you that in the mRNA, in the prokaryotic mRNA, there is Shine-Dalgarno sequence which will bind with the smaller subunit. Now in the case of eukaryotes, what's the story? In the case of eukaryotes, there is a different story. In the case of eukaryotes, you know that 5' end is having a cap, right? So that cap will help the 40S ribosomal subunit to join there. Okay, so in the eukaryotes there is no Shine-Dalgarno sequence. In the eukaryotes there will be that cap, that 5' cap will help the smaller subunit to join. Understood? Understood? This part of the diagram clear? This part of the diagram clear? And bache, here we even have something, alright, that is known as initiation factor. So in prokaryotes you have initiation factor 1, initiation factor 2, initiation factor 3. These are the 3 initiation factors that we have in the prokaryotes, right? IF means initiation factor. Initiation factor 1, 2 and 3. In eukaryotes there are factors that are written as EIFs, right? But that's not the part of our syllabus, we are ignoring it. Okay? So we have the initiation factor. So what is the first step here? The first step here is that your 30S subunit is going to join with mRNA. It is going to join with mRNA. This is the first step here, people. This is the first step here. So it will be done with the help of initiation factor 3 and magnesium ion, and you will get 30S subunit mRNA complex. Means 30S subunit and mRNA, they both will join first, and after their joining, initiation factor, right? So this initiation factor 3 is helping here. Any doubt? Any doubt? Yes. Now move to the next slide. So remember na, Shine-Dalgarno is only there in the case of okay. So now you know that, right, that here you have the start codon also, right? Right, the start codon is going to be AUG. So the charged tRNA will come up here. The charged tRNA having methionine will come up here, right? This is the second thing that is going to happen. What is going to happen? Let me write it here. We have 30S ribosome subunit. We have 30S ribosome subunit and mRNA complex. This is what we have. So this is going to join with your amino acyl tRNA. Okay, this is going to join with your what? With your amino acyl tRNA, means your charged tRNA. And for this particular step, what is going to happen, bache? Your GTP is going to play the role. Your initiation factor 2 and even 2 and 3 mainly they play the role here. And of course for this step also magnesium is required. Clear? Magnesium is required. So this is the first step. So you know that it is the amino acyl tRNA and it will be the formyl methionine. Right? I told you na, in prokaryotes it is going to be the formyl methionine. Okay. So ultimately what are you going to get after all this? After all this, another complex, amino acyl tRNA 30S subunit mRNA complex, means all these three they will join. All these three they will join. Am I right? All these three they will join. Am I right? Am I right? So obviously this is having the anticodon. It will recognize the codon here, and that's how they are going to bind. That's how they are going to bind. So I can pick up one question from this side. That question is that what is going to join first? So to that mRNA ribosome, and then to the mRNA ribosome complex, your tRNA. And now what's the next, bache? What's the next, bache? What is the next, bache? Your larger subunit will come. Now your larger subunit will also come. So it will come at the end. Clear? It is going to come at the end. Is that clear? Is that clear? Yes or no? So please remember one thing, right? That larger subunit, I told you it is having how many sites, bache? It is having how many sites, bache? It is having how many sites? How many sites are there in the larger subunit? 3 sites, right? So the larger subunit is your 50S in prokaryotes, 30S in your eukaryotes, the smaller one. So larger subunit is having 3 sites, right? What is having 3 sites, bache? Larger subunit is having 3 sites. EPA, remember the sequence, EPA. So E means exit, P means peptidal site, right? A means amino acyl site. Is that clear? Is that clear? Yes. Is that clear? Tell me, is that clear? So after this particular step, initiation factors will be released. After the larger subunit of the ribosome joins here, initiation factor will be released. Can you focus on the one point here? Can you focus on the one point here, that first tRNA comes at P site. First charged tRNA is always at the P site of larger subunit. It is always at the P site of larger subunit. Can you see that? Can you see that? The first charged tRNA is always at P site of the larger subunit. This is something very important. People, are you getting it? If it is a yes, do let me know in the chat section. Yes, if it is a yes, do let me know in the chat section. Do let me know in the chat section. Yes, 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 quick. Be quick. Yep. Clear? Sure. So this is up to this part, it is the initiation. Now let's talk about the elongation. Thus in the starting, the first charged tRNA will come to P site, right? It will come to P site of the larger subunit. Done, bache? So let me show you this. So that's E, that's P, and the next one is going to be A. So in the same way, another amino acid will charge. And how come it is possible? Obviously the codon will be read. No, the mRNA is having the codon, that will be the mRNA is having the codon. As per that information, the charged tRNA, the next charged tRNA will come up. Okay, and the second charged tRNA, it is going to come at the A site. It is going to come at the A site. Is that clear? Is that clear? Yes or no? Is that clear? Yes or no? Let's say, fine, is that clear? The next tRNA will come to the A site. Now bache, you know that the COOH group of amino acid attaches to amino acid acceptor arm. Am I right? With the amino acyl bond, this is what you know na. Here you know that amino acid is there. Here also you know that amino acid is there. So in both the cases, basically in all the charged tRNAs, it is the COOH group, right? It is the COOH group. Yes or no, people? It is the COOH group, right? That will, right? It is the COOH group. Yes, bache, COOH group that is going to form the bond here with that particular tRNA. That is what we know, isn't it? Now, what is going to happen here, right? See, this is your one amino acid. I'm taking this example randomly as of now. Hope you have seen the lecture on biomolecules. Now, what is happening here? Yes, bache, Radha Krishna, what is happening here? Now, here at P site, right? Here at P site, the COH connection with this tRNA, it will become weak, right? The COH connection of that amino acid with that tRNA, it will become weak. It's like that, no? Right? The COH connection with this tRNA, it will become weak and it will break. Right? I am talking about the P site. I am talking about the charged tRNA at P site. So here that connection will break, bache. What is going to happen here? Here that connection will break. Now because that connection will break, this amino acid is kind of free. Now because this connection will break, this amino acid is kind of free here. It's kind of free here. Yes or no? Guys, please tell me in the chat section if you are not interested in completing this topic. Let me know in the chat section. I will end the class. I don't mind in ending the class as well. Right? Please save my efforts. Let me know, please. I'll be very happy to end this class. Tell me. So I am saying that at P site there is a charged tRNA. Okay, so basically the connection here will become weak. This amino acid will get free. Now once it is free, this particular amino acid is going to form the bond with the amino acid present at A site. It's just like a baby, right? Sometimes babies, they are having their toys, right? But when they look at something else, they want that thing, right? They want that thing. It is more like this. Clear? It is more like this. Are you getting it? So this first amino acid, the connection will break. Now this amino acid is ready to form a bond with the, it is ready to form a bond with the next amino acid. So you can see this. This is the first amino acid. This is the first amino acid. This is the second amino acid. You know that how that amino acids are going to form the bond, right? Do you know about the peptide bond? Do you know about the peptide bond? This is first amino acid. So COOH of first amino acid, NH2 of second amino acid. Here, bache, what is going to happen? The water will be released, right? The water will be released. The dehydration will occur, right? And after that dehydration, this is what we are going to get. Right? This is what we are going to get. And that is, yep, that is C double bond O, NH. This is the peptide bond that we are going to get here. Can you see that? This is the peptide bond that you are going to get here. Can you see that? So basically when two amino acids will join together, this is the dipeptide. So actually what is happening, the amino acid present at P, right, it will break the connection with this tRNA. It is available for the bonding with this amino acid. Now after this, what is going to happen, bache? Please observe the diagrams carefully. It will clear all your doubts. Okay, if you will look at these diagrams carefully now, so it will clear all your doubts. So finally here you will be having two amino acids. Can you see that? Can you see that? That is going to happen. Okay, that is going to happen. This is the E side, this is the P side, this is the A side, right? So in elongation, there are elongation factors also. The amino acid present at the P side will form a bond with the amino acid present at A side. You know that which bond is going to be there? The peptide bond. Who's going to form that peptide bond? It will be catalyzed by peptidal transferase, right? But a larger subunit is having 23S ribosomal RNA. Can you see that? This is something very important. You know that this larger subunit of the prokaryotic ribosome, it is having 23S ribosomal RNA. Actually, it is the peptidal transferase. It is the peptidal transferase. You know that RNA is acting like an enzyme. So which word are we going to use for it? It is the ribozyme. Which word are we going to use for it? It is the ribozyme. What is it? It is the ribozyme. Yes, bache. What is it? It is the ribozyme. So what is happening here? The amino acid at P side will form the bond with the amino acid at A side. This is catalyzed by enzyme peptidal transferase, right? 23S ribosomal RNA is actually the peptidal transferase. It is the ribozyme. It is the ribozyme. So after this, what has happened? The two amino acid, now they come to the tRNA at A side and the tRNA at P side is free. It is empty, isn't it? It is empty. Yes or no? So now which translocation of ribosome will occur. Now what is going to happen? Translocation of ribosome will be there in 5' to 3' direction. Now it's not right. Now remember this point. Now your ribosome will translocate. Why? Because it has to read the codes present on the mRNA. Right? It has to read the codes present on the mRNA. Are you getting my point? So ribosome is going to slide over the mRNA in 5' to 3' direction. Clear, bache? Right? It is going to slide over the mRNA in 5' to 3' direction, and GTP is required for this translocation and for this sliding. GTP is required for this translocation and for this sliding. So now what is going to be there? Wait, let me show you. So after this now, because of this translocation, like it's like I'm moving a bit like this, like I'm at this point. Earlier I was at point A, now I am at point B. It's like that. So now ribosome is moving. When ribosome will move, obviously the tRNA which was at P side, it will come to E side. The tRNA which was at A side, it will come to P side. Yes or no? This is what translocation is. This is what translocation is. And who's going to translocate? The ribosome. So this is the translocation of ribosome in 5' to 3' direction. And here again I'm repeating, GTP is required. GTP is required. And there is one enzyme which is known as translocase. It is catalyzing this process, right? The enzyme translocase is catalyzing this process. Clear, bache? Translocase is catalyzing this process. So obviously look at the diagrams. Look at the diagrams. So what happened first? the smaller subunit will join with the mRNA, then charged tRNA will come, then the larger subunit will come. So the first tRNA is at the P side, next tRNA will come at the A side. So once the next tRNA come at the A side, these two amino acids, they will form a bond. That bond will be formed by peptidyl transferase, which is an enzyme which is also known as ribozyme, right? Then bache, the ribosome will translocate in 5' to 3' direction. tRNA at P site will come to E site, tRNA at A site will come to P site. Now again you can see A site is free, the next tRNA as per that codon will come, right? So that's how further the amino acids will be added, right? That is how further the amino acids will be added, and finally bache, when they will find the stop codon, the translation will stop because of the release factors. That's it, that's all. Okay, there are the release factors in prokaryotes, RF1, RF2, RF3, that will help in the release of that, right, complete complex. So once stop codons are there, you know that stop codons can be umber, ochre and opal, they will stop the translation. Release factor will release the polypeptide chain. So here there is a groove from where the polypeptide chain will come out. So that's how this process will occur. So once this empty tRNA, it comes to the E site, it will leave this, right, and then next P, again the bonding will form, and the same way translocation will occur, the same way amino acid, the tRNA will leave, and that's how the process is going to take place. Are you getting it? Are you getting it? Right? So in the case of prokaryotes, the elongation factors, okay, in the case of prokaryotes, the elongation factors are EF2, EFTS, EFG. These are the three elongation factors in prokaryotes. Okay, 2+G, 2+G, that's how you can remember, right? And finally, you know that we have the release factors. Once the nonsense codon is nonsense codon will be recognized by this ribosome, right? Release factors will release this, and GTP is again required here. Fine, GTP is again required here. So this is all about the translation. Trust me, it's very easy. Okay, trust me, it is very easy. Okay, so that is what the translation is. CEPA, that's how it will translocate, right? So empty tRNA will come out from the E side, next will come to the P side, then A. So see, this is the groove from where the growing polypeptide chain will come out. Clear, bache? So look at the diagram given in NCERT. So from the exit side, the empty tRNA will come, right? So next tRNA is here, they will form the bond, and that's how the growing polypeptide chain is coming out. So see, these amino acids are written here now. At least you should know their codons if you are targeting 360 out of 360. Okay, okay. So amino acid, you know, they will join with the help of peptide bond. Peptide bond requires energy. So in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, means through that specific tRNA, right? Right, bache? So see the diagrams, your mRNA, then how the ribosome, see, this is smaller subunit, then this is coming, that's how they are joining. Okay, can you see that? Can you see that? So you should know about the tRNA structure, and you should know about this. See, AUG start codon. Okay, AUG start codon. So in initiation, the translation, the components of translation apparatus, they come together with an mRNA, tRNA carrying the first amino acid. They will bind. Okay, see this, EPA, 3 sites are there. Next tRNA is coming to A site. They are forming the bond. Once they will recognize the stop codon, release factors will play their role. GTP will be used from this groove. The growing polypeptide will be completely released. Okay, so I'll provide you the notes in the Telegram group. You can check the images there, right? And you can use this for the revision. Fine, fine. Is that clear? So see, growing polypeptide from the exit side, these tRNAs, these free tRNAs are coming, they will be again used. Okay, so as I said about the factors, so initiation factor 2 and 3, they are present in prokaryotes, but they are not there in eukaryotes. Elongation factor 2, TU, TSG are present in prokaryotes. Release factors RF1, RF2, RF3 are present. Okay. Okay, so here also it is written RF3, it requires GTP, right? All these elongation factors, they require GTP, and here only IF2 require GTP. Okay, so it is explained with their function. That's it, that's all. So this is all about the translation. So if there is any doubt, do let me know, bache. If there is any doubt related to translation, do let me know. Quick. Do you have any doubt, or you are just here for the chit chat? But a nascent polypeptide, that newly formed polypeptide, it has to undergo many changes. It's not like that the polypeptide that is formed, that can be used as such. No, it is just the newly formed polypeptide which will undergo many changes, and finally that you will get that mature protein, the one that you need. Okay, the one that you need. So this is all about the translation, as I said. So remember this point. The ribosome consists of structural RNAs and about 80 different proteins. So it was a question in NEET 2023, right? It was a question there in NEET 2023, this line. Okay, so in its inactive state, it exists as two subunits. What? Ribosome. Okay. So see this point also, ribosome also act as a catalyst. I told you already about this 23S ribosomal RNA in bacteria, it is a ribozyme. So bache, there is no point, there is no concept that we didn't discuss so far. Okay, we have covered everything here, everything here. So you just need to, after this lecture, you just need to revise it from NCERT. That's it, that's all. Okay, chalo, now let's move to the next part. Again, as per NCERT, it is written that a translational unit in mRNA is the sequence of mRNA that is flanked by the start codon and stop codon and codes for polypeptide. Remember that mRNA is having that, no? Start codon and the stop codon, and you know that in between we have the UTRs also, right? So this is the one which will be translated. This is the one which will be translated. Clear, bache? Clear, bache? So UTRs, they are present. You know that they are required for efficient translation process, right? This is the role of UTRs. Efficient translation, they efficient the translation process. Okay. Okay, so do read it from NCERT. Trust me, you are going to find it very easy. And let's focus on the next topic, that is the regulation of gene expression. Basically, we are going to talk about your operants, another important and the simple topic. Okay, so we have two options. We can take a break for 15 minutes and then we can continue. The another option is ki let's finish the chapter in one go. I think till 11, then we can finish this chapter. What do you say? Right, if we without break we can directly proceed and we can literally finish this chapter till 11, right? It's very simple. Lac operon will hardly take you 30 minutes. Then after that, genetic code is purely theoretical. We have to human genome project is purely theoretical. We just need to follow the NCERT, and then comes the DNA fingerprinting. So what say? Let's finish it now. Let's continue. है ना? Let's continue. Chalo. So next topic is the regulation of gene expression. What is the next topic that we have? Right, it is the regulation of gene expression. Array. What is it? It is the regulation of gene expression. That how can we regulate, how can we control the expression of a gene? So I am telling you this thing again, bache, how can a gene expresses itself? A gene is going to make the protein ultimately, right? By making the protein, it expresses itself. So if we want to regulate that expression, na, in the case of eukaryotes, there are many steps. Why? Because in the case of eukaryotes, they are having a proper nucleus. So within the nucleus, transcription is there. So at that level we can control the expression. RNA processing is there. At that level we can control the expression. We can remove a particular intron. Sometimes we also remove some exons. We join exons in different ways. We can get different proteins. So at that level also we can regulate. At the level of translation also, and when we move that RNA to the nucleus, at that level also. But in prokaryotes, it's not like that. You know that transcription and translation, it is moving simultaneously. So because of that thing, bache, because of that thing in the prokaryotes, right, we do not have many levels of regulation. Just have a look. In eukaryotes, regulation could be exerted at transcriptional level, processing level, transport of mRNA from nucleus to cytoplasm level, and at translational level. But in the case of eukaryotes, it is not like that, right? In the case of prokaryotes, it is not like that. So in the case of prokaryotes, what are we going to talk about? We are going to talk about the operons. So let me tell you, the concept of operon is only there in bacteria, only there in prokaryotes. It is not there in eukaryotes. जैसे RNA processing is only there in the eukaryotes, it is not there in prokaryotes. Same way, the operon concept is there in the prokaryotes only, it is not there in the eukaryotes. Clear, बच्चे? Is it clear? Sure. So now you know that we have two type of genes. One is constitutive genes. Yes or no? Do you know that? What type of genes do we have? Is it constitutive? Is it constitutive? Constitutive genes and non-constitutive genes. Right? So we have two words. What do we have? Constitutive genes, non-constitutive genes. Constitutive genes, we have non-constitutive genes. So constitutive genes means they are housekeeping genes. What are they, people? They are housekeeping genes. And what about the non-constitutive? They are the luxury genes. Right? They are the luxury genes. So now you please listen to this concept very carefully. That's important and a very simple concept. Trust me, operon topic is one of the simplest topics. Right, bache? So please pay attention. One is constitutive, another is non-constitutive. Constitutive, another is non-constitutive. Constitutive, housekeeping. They are always active. We always need their expression. Right? We always need their expression. We always want these genes to make proteins. What is the meaning of their expression? Means genes are going to form the protein. So we always need these genes to make the proteins. Now, luxury genes. The word is luxury. So obviously you can relate. We don't need their expression always. Right? They are switched off. We need to switch on them when we need their expression. Now here, when you talk about the housekeeping genes which are always open, like genes for glycolysis, we always need the glycolysis to be there in our body. Right? You know, na, glycolysis, glucose will form 2-pyruvate. Right? That is the very first step of respiration. So genes for glycolysis, the enzymes for glycolysis, they are always required. So that genes are always giving their expression. Even the genes for your ATPase, right? The genes for your what? The genes for your ATPase enzyme, they are always active. They are always active. Now when it comes to the non-constitutive genes, also known as smart genes, right? We normally, by default, let's say they are off. So we have to switch them on when we need them. Right? When we need them, what we need to do here? We need to switch them on. Right? What we need to do? Bache, we need to switch them on. Is that clear? Is that clear? So by default they are off, we have to switch them on. Right? So basically their expression is not always there. Okay. Yes, bache. Their expression is not always there. Sometimes, right, when we need their expression, we have to switch them on, and when we do not need their expression, we have to switch them off. Okay, so we need their expression when required, otherwise we will switch it off or switch it on. Is that clear? Right? So here in these non-constitutive genes, we will be discussing the concept of inducible and repressible. So now let's understand that terminology. Inducible, to induce, means somewhere to start, isn't it? This is the meaning here. Repressible, to repress, to stop something, right? To stop something. Now let's say you entered in a room. Okay, let's say you entered in a room, the light is switched off. What will you do? You will come and you will switch on the light, right? When it is the inducible, When the word is inducible, right? When the word is inducible, what is there? Inducible means to start. So if you want to start something, what you have to do, right? First of all, obviously that thing need to be in the switched off condition, then only you can switch it on now. So by default, can I say that? That by default it is switched off. By default it is switched off, so you have to switch it on. And here by default it is switch on and you have to switch it off, right? That's how it is going to work. That's how it is going to work, yes or no? That is how it is going to work, yes or no? Yes or no? है ना? So that is what we need to discuss. So the very first thing is, what actually is the operon, right? What actually is the operon? I told you now, operon is this concept we are only going to read in the case of prokaryotes. It is not there in the eukaryotes. So what is operon? First of all, tell me this. But it's very simple that in 1961, right, two French microbiologists, right, their name is Jacob and Monod basically, right? Jacob and Monod, Francis Jacob and Jacques Monod, right? So in E. coli, obviously E. coli is very famous, you know na? Our E. coli is very famous, do you know that? Yes, E. coli is very famous. So basically they proposed the operon model for the regulation of gene expression in E. coli, right? So in 1961, two French microbiologists, Jacob and Monod, in E. coli, they proposed operon model, yes, to regulate gene expression, okay, to regulate gene expression. So now bache, before starting it na, we should know what exactly is the meaning of operon, right? That is what we should know. Actually, operon is a group of genes, okay? It's a group of genes which is having one regulatory gene. When the word is regulation, to regulate something, to control something, it's not always means that we need to switch off something, right? It can be anything as per the demand. Like let's say you are an officer, right? There is a chaotic situation, you will go there, you will regulate, right? You will regulate that situation, you will check what is the issue basically, okay? Accordingly you are going to take the action, isn't it? So regulation is not always mean in a negative side, you have to stop something. No, as per the need, maybe you have to start something, maybe you have to stop something, right? So now when you are talking about the operon is nothing but it is a group of gene, right? It is a group of gene having one regulator, having common promoter, one operator, and there are many structural genes, right? There are many structural genes. Mr. Zain, you are already not studying here, you are here for the chit chat, then why are you spamming here for the break? Okay? You don't have anything else to do. Although you people are that free. I think you seriously, you're not here even to study, nothing, but you just need to spam here. Like you people are that lonely, you do not have physical friends, you use online platform for chit chatting and all, you don't have any member in your family, kuch bhi. So operon is the word. What is the word? Operon is the word. Okay. Okay. So as I said, what is an operon? It is a part of the DNA where there is a single regulatory gene and having more than one structural genes, operator, a promoter and a regulator. Okay. Operon is a single regulated unit. It is the, is the part of DNA having single regulated unit containing one or more than one structural genes. Right, one or more than one structural gene, one promoter, operator, and regulator gene. It's a single regulated unit. Like it works all together, right? It works all together, isn't it? What is it? It is a single regulated unit, right, having one or more structural genes, operator, promoter, and a regulator gene. So it works all together. Is that clear? It works all together. Is that clear? This is the meaning of operon. And here in the operon, we will be discussing two things, right? One is the inducible operon and another is the repressible. You know the meaning of inducible. Means to start. Repressible means to stop. So in inducible operon, you will talk about the lac operon of E. coli. This is what we are going to discuss. Lac operon means, bache, lac means lactose. It's a disaccharide made up of glucose and galactose. And in repressible operon, we have the example of tryptophan operon. Bache, tryptophan is what? It is an amino acid. Right, tryptophan is what? It is an aromatic amino acid. Is that clear? So this is what we have. So in detail in our syllabus, we have inducible operon, right? We have inducible operon in our syllabus in detail. Is that clear? Right, what do we have? We have inducible operon in detail in our syllabus. Clear? So inducible operon means to start gene expression. Right, here what are we going to do? We are going to start gene expression. Clear? We have to start the gene expression. So let's take the example of course, Escherichia coli, that is your E. coli. Normally, bache, E. coli feeds on glucose. Right? Mani, Princess, Chetu, Lochan, everyone here in the class. Normally, E. coli feeds on what? It feeds on glucose. Right? It feeds on what? It feeds on glucose. Right? Right? So if it is getting the glucose, it will take it, it will digest it. Now let's say I am growing E. coli in a medium and instead of glucose, right? Instead of glucose, I am providing that E. coli the lactose. Right? Again, I am repeating this point. Normally, E. coli feeds on glucose. Normally, E. coli feeds on glucose. Now, let's say I change the source of food. Okay, I change the source of food. Instead of glucose, I start giving it, right? I started giving it lactose, right? What is a lactose, bache? Lactose is a disaccharide. I hope you remember it's a milk sugar. It's a milk sugar. It's a disaccharide. It is made up of glucose and galactose. You remember? It is made up of glucose and galactose. So what I just did, I changed the source of food for the E. coli. Now E. coli is like, here I used to digest glucose, but now I have to survive, right? So somehow I have to start digesting the lactose as well. So in E. coli, right, when you talk about E. coli's DNA, it is having genes for the digestion of E. coli, for the digestion of lactose. Do you know that? In the E. coli, in its genetic material, it is having the genes which can digest the lactose, which can break the lactose, but they are by default off. So because it is coming under the example of inducible operon, means we have to start their expression. So if you want to start the expression, firstly you should understand what went wrong, why that lac operon is not activated. है ना? Why that lac operon is not activated, why that lac operon is not giving its expression, why that genes for digesting lactose, they are not active. This is what we need to understand. Let's understand this. So everyone in the class type one thing. Everyone say peepo. Everyone say peepo. Okay? So whenever you are going to click the selfie or the picture, you are not going to say cheese. Okay, it should be peepo. Okay, it should be peepo. What should be there? Peepo. So let me write that peepo. This is P, this is I, this is again P, this is O, and then that will be Z, Y, E. Then there will be Z, Y, E. then there will be ZYA, it is people. So we are going to start our story from the I. This I is actually showing us the regulator gene or regulatory gene. This P means the promoter here. This O means the operator here. Clear bache? Clear bache? And this ZYA, they are what? They are the structural genes. What are they? They are the structural genes. Are you getting it? What is P here? P is promoter, O is operator, I is the regulatory gene. Right? P is promoter, O is operator, I is the regulatory gene. Right, bache? Right, promoter, operator, regulatory gene. This is what we have. So now when you talk about this I, right, as I said, it is the regulatory genes, means it is doing something with the regulation of this DNA segment. It is doing something with the regulation of this particular operator. Am I right? Am I right? So bache, I here used to make mRNA, of course, and that mRNA is making a protein. है ना? This protein is actually a repressor protein. This protein is actually a repressor protein. Which protein it is, Parul? It is a repressor protein. And this repressor protein, it get attached to the operator. This is the default condition. This is the default condition. This is by default. Everything is happening here by default. Here the by default expression is not there. It is switched off. So what is happening here in switched off condition? This regulatory gene has formed the mRNA, which is transcription, which has formed the protein, that is translation. So actually the protein formed here is a repressor, and that repressor will attaches itself to the operator. So can I say one thing here? Can I say one thing here, that product of regulatory gene, this is NCERT language bache, can I say one thing here, that product of regulatory gene interacts with operator, can I say so? Product of regulatory gene means the protein that is formed by this regulatory gene. It interacts with operator means it attaches itself to the operator. So product of regulatory genes, it interacts with operator and it regulates the expression here. Can I say so? It regulates the expression here. Yes, people tell me, can I say so? Can I say so? Everyone in the class. The product of the regulatory gene is interacting with the operator, and that is basically regulating the expression here. Actually, bache, remember one thing, right? Do remember one thing, that promoter and operator, they are overlapping. Promoter and operator, this region is actually overlapping, right? This region is actually overlapping. Clear? Because repressor, you can write down, because repressor is attached to operator, so RNA polymerase cannot bind to promoter. RNA polymerase cannot bind to promoter, and transcription translation cannot start. Right? It cannot start. Are you getting it? I'm repeating my points again. My first point is product of regulatory gene means the protein formed by regulatory gene. The protein formed by regulatory gene, it binds to, yes people, please tell me clearly. Yeah, please type in the chat section. It is going to bind to, it will bind to operator. Whatever regulatory gene is going to form, it will bind to operator. Now your RNA polymerase. What's next? RNA polymerase. What is next? The next is RNA polymerase. P for polymerase, P for promoter. RNA polymerase always binds to promoter. So please do not make the mistake here. Right? RNA polymerase, it always binds to the promoter. Clear, bache? Yes बच्चे. So product of regulatory gene, it binds to operator. RNA polymerase, it binds to promoter. Remember this thing. And you know that in the lac operant, these are the three structural genes that you have. So Z will give you beta galactosidase. This is the enzyme that you are going to get from Z. From Y, you will get permease. And from A, you will get trans-acetylase. So that should be there in your notes, right? This should be in your short notes. You should include such points in your short notes, right people? You should include these points in your short notes. Can you see that? So product of regulatory gene will bind to operator. The RNA polymerase binds to promoter. Z is going to give you beta galactosidase. Y is going to give you permease. A is going to give you trans-acetylase. Right? I'll tell you the function here. Okay, I will tell you the function here, but tell me up to this part all clear? Yes? Done? So by default, that's the scenario. The scenario is, by default, that's the scenario. The scenario is regulatory gene is forming a protein which is attached to the operator, which is not letting RNA polymerase to bind to the promoter. That's why transcription translation is not there. So one thing is clear, bache, that if I have to start, or if I want to start, okay, if I want to start the transcription translation here, I cannot allow the repressor to get attached. I cannot allow the repressor to get attached to the operator, isn't it? I have to stop the repressor to get attached to the operator, isn't it? Isn't it? Isn't it? Yes or no? I'll tell you one story here. Bache, your Z, I told you it will give you beta galactosidase. Now this beta galactosidase, it is very important because this enzyme will act on lactose. This beta galactosidase, this enzyme will act on lactose. It will break that lactose into glucose and galactose. It will break that lactose into glucose and galactose. And moreover, the Y will give you permease. Do you know the role of permease? The role of permease is to allow lactose within E. coli. It will allow lactose within E. coli. It will let lactose to enter within E. coli. A is for trans-acetylase, right? A is for trans-acetylase, right? So A is for trans-acetylase. So it transfer acetyl group of coenzyme A to beta galactosidase. Basically its function is not so clear, bache, but it is somewhere dealing with, you know, removing the toxic material or something. Okay, so it is going to transfer its acetyl group from acetyl coenzyme A to beta galactosidase. This is the role of these three enzymes. This is the role of these three enzymes. So now if I want, right, if I want the genes to give their expression, so I have to stop this repressor to bind to the operator. Am I right? Right? I don't want, because I want to switch on the expression here, right? I am studying the inducible operon, right? I am studying the inducible operon. And do you know what exactly I am studying, students? Do you know what exactly I am studying? Yes, people. Do you know what exactly am I studying here? Anyone? It is the negative regulation of the, right? It is the negative regulation or negative control of inducible operon. Do you know the meaning of this negative regulation? I will come to this after 1 minute, right? I'll explain it after 1 minute. You please pay attention. I'm saying inducible operon. I'm telling you here, if you want to induce the expression, you cannot let your repressor to bind to the operator. This is something that I don't want. So basically I have to distract this regulatory protein, this repressor. I don't want this repressor to get attached to what? To get attached to the operator. Now what can I do? I need to distract it. How can I? Let's say if your friend is not interested, if your friend is like, I don't want my friend to score good marks, what are they going to offer you? They can offer you the video games, they can offer you the good magazines, they can offer you their Netflix password, Amazon Prime password, this and that password. You see, you want to watch this movie, array, this movie is very good, I'll give you my password, you watch it. right? You go, you do this, right? So that so obviously if you will watch a movie, you will not give time to your study, right? You will be distracted. Yeah, it is peepo. Yes, bache, it is peepo. It is peepo na. Here I missed it. It's okay, I'll add it. Thanks. Hai na, this is what your friend is going to do with you. This is what your friend is going to do with you, right? Yes or no? Isn't it? Isn't it? So same is the case here. We need to distract this repressor, and for that what can we do? We should give this repressor a distraction, and for that repressor, okay, let's say so we need to distract this repressor, and that will be done with the help of inducer, right? And that will be done with the help of inducer. And do you know that what is that inducer? The lactose itself. The lactose itself. Actually, its isomeric form allolactose is allolactose will form here. Actually, that is the true inducer, right? But you can also go with the lactose. That's fine for your NEET UG. Okay, so the lactose will come. Lactose will be like, "Arey yaar, why are you going to that operator? That operator is so boring. You come to me na. You come to me. We will go out. We will watch movie. Hannah, we will do this. We will do that." So what is going to happen once the inducer is there in the system? That inducer is what? That inducer is lactose. That inducer is what? That inducer is lactose. So when lactose is there in E. coli, it will bind with that repressor. So now repressor will not be able to bind to the operator, right? What will we get? We will get inducer repressor complex. We will get inducer repressor complex, and that inducer repressor complex, right, now it has formed. So repressor will not be able to bind with the operator. So now RNA polymerase is very smart. That is going to take the advantage. So because of that, RNA polymerase binds to promoter, right? So when operator is free, when operator is not attached with the repressor, RNA polymerase binds to the promoter. This results in starting the transcription of your ZYA, and finally you will get all that enzymes. That is how the switching on of the expression will be there, right? And that is how the inducible operon will work. Just have a look. Just have a look. So this repressor, it attaches itself to operator. Because of that, RNA polymerase cannot bind to the promoter, and the transcription translation is not there. Now when this repressor protein, when inducer is there, what is inducer? The inducer is lactose. So when this inducer is attached to this repressor, repressor will become inactive, right? Repressor will become inactive, right? And RNA polymerase will attach to the promoter. Because of that, RNA polymerase binds to the promoter. So once RNA polymerase binds to the promoter, bache, the ZYA, they will be transcribed. You will have this like mRNA. You will get these 3 enzymes, right? And then the lactose will be digested. Isn't it? That is how it works. That is how it works. Clear? But now here, no one asked me. There is a question mark. Ma'am, you said that it is the permease that allow lactose to enter within cell. Right? Permease allow lactose to enter within bacteria. But you are saying that, hai na? So now here, no one asked me, ma'am, if permease is allowing the lactose to enter within the E. coli or within the bacteria, then how come lactose can act like an inducer? Hai na? Hai na? How lactose can be the inducer? How is it possible? It's very simple, bache. Always remember, some expression of permease is always there, right? Some expression of Y gene is always there. It's not like that permease is always absent in E. coli. No, it's some expression will always be there. Very minute expression will be there, and because of that, lactose will, even the very less amount of lactose will enter within E. coli. That lactose will act like an inducer, and further it will start transcription translation, and then obviously then the lactose entry will be easy. Okay, so always remember, some expression of Y gene is always there. Okay, some expression of Y gene is always there. Is it clear? Yes, is it clear? Sure, sure बच्चे. Some expression of Y gene is always, it is always there. Okay, I hope it is clear now, right? I hope it is clear now. So in the paper they can ask you the questions on the basis of that, right? Right? They can always ask you the question on the basis of what? They can ask you the question like regulatory gene, the product of regulatory gene, where will it bind? Where that RNA polymerase bind? What if after the beta galactosidase there is a, after the Z gene there is a stop codon? What is going to happen? The permease will not form. No permease means no lactose entry, means no transcription, translation. That's what you need to remember. Now bache, I just use the word negative regulation, right? What you just studied in your syllabus, you studied, right? You studied the inducible operon, right? But the negative control of inducible operon or negative regulation of inducible operon, that's what we have studied. What we have studied? We studied the inducible operon. Inducible mean to switch on, right? But the negative regulation, this is what we have studied. Or you can say that negative control of inducible operon, that's what we have studied. So now what is the meaning of that? See, if your regulatory gene, okay, if your regulatory gene, if it forms repressor, right, this is something very important. And let me tell you here also, in this particular topic also, I have seen many teachers making mistakes, right? So please pay attention. So if regulatory gene is forming a repressor, right, if it is forming a repressor, then always it is going to be the negative control, or always it is going to be the negative regulation. Okay, if your regulatory gene is forming the repressor, and if your regulatory gene, if your regulatory gene is forming the activator, then bache, what is it? It is the positive regulation. Always remember it. Then it is what? It is the positive regulation. Are you getting my point? Then it is the positive regulation. Means if regulatory gene is forming the repressor, it is the negative regulation. If regulatory gene is forming the activator, it is the positive regulation. And in your syllabus, right, even if you talk about the tryptophan operon, you have negative control only. Why? Because regulatory gene is forming the repressor. So yes, inducible operon can have two control, positive and negative, but in your syllabus only negative control is given, right Lochan? In your syllabus only negative control is given. Is it clear? Is it clear? Sure? Sure. So both are possible, negative regulation as well, positive, but in our syllabus we just have the negative control. Okay, we just have the negative control. That's the point. Understood? That's the point. Fine. Fine. So this is all about the inducible operon, right? And one student is also asking about that CAM, the catabolite. Yes, about that catabolite expression and all, right? That is not the part of your syllabus, bache, that's why I am not discussing it. Now there is one more operon, not the part of your syllabus, but yes, you should know it a bit. So this is the tryptophan operon. Tryptophan operon is also the example of the, it is the repressible operon. It is the repressible operon, and again we are going to discuss the negative regulation of this repressible operon. So what is the meaning of negative regulation? Negative regulation means the regulatory gene is forming the repressor. Negative regulation means, Lochan, the regulatory gene is forming the repressor. So it's a repressible operon. What we have to do, we have to switch it off. So basically, can I say that by default it is switched on? Can I say that by default it is switched on? Yes, obviously. So here what is happening, that this regulatory gene is forming the mRNA, that is making the repressor, right? It is making the repressor. But the point here is this is inactive repressor. This is not something that can bind to the operator. This is inactive, right? This is inactive. Clear, bache? So that is why here the polymerase is attached, and we are getting, we are getting the transcription and the translation is there. Okay, so what can we do in this scenario? Parul bache, what can we do in this scenario? We have to make this repressor active. So here what will come? Your tryptophan. Your tryptophan act like a co-repressor, because this tryptophan is already too much in the E. coli's body, in the cell, right? This tryptophan is like, okay, my expression is too much, now it has to stop. Okay, it has to stop. So what is going to happen? The tryptophan will act like a co-repressor, right? That tryptophan will come here, it will bind, like this is the repressor here, this is the co-repressor here, this is the repressor here, this is the co-repressor. They both will, when this tryptophan will join with this inactive repressor, wait, let me write it. So inactive repressor plus co-repressor. What is that co-repressor? That is also your tryptophan, right? So that will make active repressor, and that active repressor will join with operator. So once it will join with operator, you know what will it do? Once it will join with operator, it will stop gene expression. It is going to stop gene expression. Understood? Understood? So this will join here to the operator, right? And then it will stop the expression. That's how it works. Okay? That's how it works. Understood? Understood? Sure? Okay, so this is all about the gene expression in your prokaryotes, right? This is all about what? All about the operons. So the next topic, second last topic that we need to discuss is HGP, that is your Human Genome Project. So students, if you want a break for 10 to 15 minutes, I can give you, but then let's finish this topic. What say? It is for Lochan, yes, you only need NCERT for it, and for sure one question will come from HGP, one from DNA fingerprinting, one from genetic code, right? This time you are going to get 6 to 7 questions from molecular basis of inheritance. You're literally going to get 6 to 7 questions from molecular basis of inheritance. So do you need break for 10 minutes? Chalo, it's 10, let's start it. Break time. Okay, let's start at 10:15 p.m. and after that till 11 we will finish this. Okay, you know that HGP is not lengthy. Okay. Hi everyone, so are you people done with your dinner and everything? Yep, all good? Okay, so now let's continue. Okay, let's continue the session and let's talk about the Human Genome Project. Again, I'm repeating, you will for sure, you people are going to get one question from this particular part. So everyone, yes, I really want to see some energy in the chat section. Quick, everyone, guys. Vardhan, thank you so much for such appreciation. Thank you so much. I don't think so, but okay, thank you. So guys, everyone, quick. All of you, quick. And if you're new to our channel, you know what you have to do. You have to subscribe our channel. And tomorrow at 11 AM, let's practice the last 10 years PYQs of the chapter, Molecular Basis of Inheritance. And this is the coupon code that you have to use while entering there in that class. Okay, let's solve the PYQs tomorrow morning. Done, bache? So the topic is Human Genome Project. It is a mega project. And do you know why do we call it as a mega project? Because in this project, the you can say that the man force, the funds, and the duration, it was too much. Like many people, they were involved in this project, right? When it comes to the funds, bache, they are also so many. And what is the main purpose of this Human Genome Project? To sequence our DNA. Okay, to sequence our DNA. Okay, what is the meaning of sequencing our DNA? Right, you know that when it comes to the you know that our DNA is basically having what? It is having the basis, right? It is having the basis. That is what our DNA is having. Yes, people, the basis, the nitrogenous basis, the nucleotide, obviously they are present in a proper sequence, and that sequence is further coding us that protein and everything, right? So when it comes to the HGP, Human Genome Project, so scientists, they decided that we should decode, we should sequence, we should write down all the bases in a proper sequence which are present on DNA, so that they can use that information to treat the disorders. They can check, like, if I'll give you one example. Let's say here we have our Chintu, right? Chintu is perfectly healthy, right? Chintu is perfectly healthy, right? Scientists, they have sequenced the Chintu's genome. Now we have Pintu, okay? We have another person. That person is what? That person is Pintu. Now in the case of Pintu, the things are not good, right? Pintu is having some disorder, and I really want to check that what kind of disorder is there, right? What kind of disorder is there? So what will we do? Now our Chintu is healthy, I will check Chintu sequence. My Pintu is not healthy, I will check Pintu sequence. I am going to figure out the differences, right? I'm going to figure out the differences, and that differences will tell me that okay, in Pintu's sequence, that is the problem. This particular base is absent, or you can say that replaced, or whatever mutation it can be, and it is resulting in a particular disorder. So it is important, right? This project is very important. After this project, we got to know so many things. Like we got to know that in humans 99.9% DNA is same. There is a difference of only 0.1%, and you know we use that information for DNA fingerprinting. So we got that technique, and you know the benefits of DNA fingerprinting as well, right? For solving the crime, for solving the paternity-maternity issues, right, bache? Now, because of this HGP, we got to know that in our DNA, only less than 2% is providing us the information for the protein. Only less than 2% is telling us, it's giving us the information for that proteins. Right? Imagine, this is what we got to know. With this Human Genome Project, bioinformatics came into origin, okay, which is another field, which is another science. Even you can have career options there, right? You should check about this. Okay, even not just the human genome, right? Scientists, they sequenced another model organisms DNA as well, right, bache? They used it for studying different things. Okay, so basically in HGP, we will be talking about the sequencing method. We will be talking about the Frederick Sanger there. Okay, Frederick Sanger there. So Sanger sequencing techniques, they were used. So in this topic HGP, we have to cover everything, and everything is important, right? You please open your NCERT. And please highlight the important point. So as I said, it was a very ambitious project of sequencing human genome, and it was launched in 1990. Important MCQ. It was launched in 1990. Next, it was called a mega project. So here you can see, you can imagine the magnitude and requirements, right? So you know that if you talk about the haploid DNA content of human, it is 3.3 into 10 raised to power 9 base pairs. Yes, bache, it is 3.3 into 10 raised to power 9 base pairs, right? And here you can see human genome is said to be 3 into 10 raised to power 9 base pair. The word is genome, right? And let me focus here, bache. Genome is always the haploid content. Do you know that? Genome is always the haploid content. Jaise, see, you know that we have 46 chromosomes, isn't it, Vardhan bache? How many chromosomes do we have? 46. Basically 23 pairs are there. One chromosome is from mother, one chromosome is from father, but right, they are the homologous chromosome. They are the homologous chromosome, Leharika. So if they are homologous chromosome, they will have same genes, same gene at same locus. The allele can vary, but the gene is going to be same, right? The gene is going to be same. This is what we know. Isn't it? Isn't it? This is what we know, right? So there is no need to study both the chromosomes, right? If we are studying the haploid content also, we will get to know about the genes. We can sequence, we can figure out their position, right? We can check the number of genes present on this chromosome, that chromosome, right? The only point is that we females are having XX chromosomes. Our sex chromosomes are also same. In males it is XY. So in the males, even we need to study the Y as well. So basically 24 chromosomes of humans, they will be sequenced, they will be studied. Any doubt here? So for this 3.3 into 10 raised to power 9 base pairs, the word is base pair, not the base. Please pay attention, right? The cost of sequencing required is $3 per base pair. So estimated cost, or you can say that it was $9 billion. So imagine the funds here, right? The requirements here. Okay, so if we have to store the obtained sequence, so we need so many books. Okay, that is not possible. Humanly, it is not possible to note down all the information that we have in our DNA. So because of that, right, what happened, bache? Right, a new science, a new area in biology, bioinformatics, it evolved with this Human Genome Project, right? With this Human Genome Project. Are you getting it? Bioinformatics. It is also a PYQ. So now the next point is goals of Human Genome Project. That what are the goals of this HGP? So you know that first thing is to identify all the approximately 20,000 to 25,000 genes in human DNA. I am clarifying this thing. These are the goals, right? These were the kind of assumptions. But after studying the human genome, after sequencing the human genome, scientists, they got to know different things. Right? They got to know different things. Right? They were assuming that we have, right, approximately 20,000 to 25,000 genes. Right? This is what, right? You can see here, this is what they assume, that scientists, they assume that they are having 20,000 to 25,000 genes, right? 20,000 to 25,000 genes. And then their goal was to determine the sequence of 3 billion chemical base pair that make up the human DNA, then to store this information in databases, then to improve tools for analyzing that data, right? And then to transfer the related technology to other sectors like such as industries. Okay, and their one other goal was to address the ethical, legal and social issues that may arise from this project, right? So these were the goals, and this is what you need to remember. Clear, bache? Clear, bache? So Human Genome Project, it was a 13-year project coordinated by US Department of Energy and National Institute of Health. Right, US Department of Energy and National Institute of Health. And during early years, the Wellcome Trust UK became the major partner here. Then bache, additional contribution came from Japan, France, Germany, China and others. So basically, you know that the funding, right, it was obtained from the different countries, and obviously it was a mega project. It took 13 plus years to decode the human sequence, and still the function of many genes, it is unknown. Do you know that? Do you know that? Right? So the project was completed in 2003. So knowledge about the effects of DNA variation among individuals can lead to the revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders. Because we know the details of the sequence, okay, we can figure out what type of mutations are causing particular disorder. We can figure out their root cause. We can stop that disorders also, right? We can stop that disorders also. Clear bache? Clear bache? Yes. So besides providing clues to understanding human biology, learning about non-human organisms, DNA sequence can lead to an understanding of their natural capability. So basically bache, we can study other organisms, we can see how can we use them for improving our health care, for environment and everything. Okay, so from this part also the question can come in the paper. They can ask you that which organisms other than human, which organisms were sequenced. So this part, Samiksha, it is also important. Okay, it is also important. Is that clear? Lochan, Samiksha, Girija, Chetu, Krutika? So bacteria, right? Bacteria, yeast, C. elegans, which is a free living non-pathogenic nematode. So from this line also the question can come, isn't it? From this line also the question can come. Which one is a free living non-pathogenic nematode? C. elegans, Caenorhabditis elegans, right? Caenorhabditis elegans, C. elegans it is. Then Drosophila, right? The plant, like your Arabidopsis, they were also sequenced. So this information is important. It should be there in your notes, right? You need to revise it. Okay, you need to revise it. Now come to the methodologies. Now see, it is not possible to sequence the entire DNA because of the technical limitations. It's not possible to isolate the entire DNA and to sequence it. So for that, what we need to do first, we need to break the DNA into the segments. Yes or no? Chetu bache, what we need to do? We need to break that DNA into the, yes everyone, we need to break that DNA into the, just a minute, yes, we need to break that DNA into what? Into the, yes everyone, please tell me, into what? Into the fragments. Okay, into what? Into the fragments we need to break that DNA. Even that is important to know. Okay, that is important to know. So basically the DNA fragments, some of them are having larger sizes, some of them are having small sizes. Okay, so we also use the vectors here in Human Genome Project. Vectors, you know na, they are the gene taxi. After this chapter, you know, next chapter that we are going to start is your, yes, the next chapter that we are going to start is your this biotechnology. You can see this, right? You have your Wasim sir's class, then your Pankuri ma'am's class, and then again the biotechnology principles and processes. So if you didn't like the session yet, please go and like this, right? So on 12th of January at 5 p.m, basically after 3 days we are going to meet again and we will complete this chapter. This is another important chapter. So can you see the sequence? We started with the human reproduction, reproductive health, then we moved to the principles, then to the molecular basis, and then to the biotech. So we are targeting the most important parts first. Okay, after this we will go for human health and disease, we will go for microbes part, right? And then your Pankuri ma'am will start the ecology part, then we will move to class 11 syllabus. Okay, so quickly everyone, if you didn't like the session yet, go and like this session. Okay, so see you after 3 days, and tomorrow I want you people to be there in my special class, which is at 11 a.m. Let's solve the questions, bache. Okay, so you will have better understanding of vectors, RNA processing in that biotechnology chapter. So just have a look. Vectors, right? They are the gene taxi. We use vectors for copying a desired gene or the to make it the multiple copies or to clone the desired gene. So in the HGP, 2 vectors were used. One was BAC, another is YAC. It is another important question. Okay, so BAC means bacterial artificial chromosome. YAC means yeast artificial chromosome. Right, so why these vectors were selected in Human Genome Project? Because they can also clone the DNA having the large size. Okay, जैसे if you talk about the plasmids, they can just copy the DNA which is having, you know, few base pair in kbs. So they can even copy the DNA having more base pairs. Okay, so that's what you need to remember. So now what they did, because we have to go for the sequencing, right? There should be a proper methodology. So when it comes to the methodology, this is what we discuss: ESTs and sequence annotation. ESTs and sequence annotation. Is that clear? EST and sequence annotation. Expressed sequence tags. Two approaches are there. First is, like, let's say this is a cell. Its DNA has formed the mRNA. DNA has formed the mRNA. You know that it means that mRNA is going to form the protein. Am I right? Am I right? You know that this is the gene expression. This is how a gene is going to express itself. It will form mRNA. mRNA will form the protein. That's what we know, people, right? So in expressed sequence tags, the genes that has expressed itself, right? The gene that has expressed itself means that has formed the mRNA. A gene that has expressed itself means the one which is making protein, the one which has expressed itself. So DNA is going to form mRNA, mRNA is going to form the protein. So here in this express sequence tags, we will just pick up the genes that are expressed as RNAs, right? So here we are not going to sequence the entire genome, right? We will just sequence the mRNAs. We will just pick up that DNA that has formed the mRNA. Okay, that has formed the mRNA only. This is what we are going to do, because it will tell us about the DNA which has expressed, okay, this thing. Sequence annotation, the whole set of genome. Here we are not considering only the mRNA part, right? We are considering the entire genome. It will include coding part as well as non-coding part. Can you see that? Whole set of genome that contain all the coding and non-coding sequence, and then later assigning different region in the sequence with function. So here they sequenced the entire genome, then they figured out, you know, certain repeated bases. They used that information and then they sequenced it. They assigned different region in the sequence with their functions. So these are the two methodologies. So question can individually come from the methodologies as well. And as I said, which vectors will be used, right? I told you, the entire sequence, the entire DNA cannot be sequenced. Technical limitation is there. So we have to take the DNA, we have to break it in fragments. We are going to use the vectors, we are going to clone that, right? And then we can sequence it. So these are the vectors. So this slide is important. You should know each and everything here. Okay, right? So as I said, DNA is isolated, converted into fragments of smaller size. They were cloned in the suitable vectors. Okay, so these are that vectors. Important MCQ. Understood? So fragments were sequenced using automated DNA sequencers that worked on the principle of method developed by Frederick Sanger. So Frederick Sanger is no one, obviously he was a scientist, right? But he was a scientist. Okay. So he made some sequencing techniques by which automated DNA sequence, basically that fragments, they were sequenced automatically. So this line is important. Clear bache? Right? And Sanger, it is given in NCERT, he got the credit for sequencing, right? For sequencing the amino acid in protein. He also got the credit for sequencing the amino acid in protein. Clear bache? So this is given in NCERT, and yes, of course, you people should know about it then. है ना? You people should know about it then. Understood? So these sequences, as I said, they were arranged based on some overlapping region present in them. Try to understand this. Try to understand it. We have the human genome. I told you less than 2% of the human genome will give us the functional information. Rest all is non-coding. Now we are taking its fragments. So when we are taking its fragment in our genome, right, this is what you should know. We used to say 99.9% we have same DNA. The difference is of only 0.1%. That's what I am telling you. That 99.9% we are all same. The difference is of only 0.1%. Right. So basically in our case there are certain overlapping regions. That overlapping region somewhere, right, that snurps. okay, that SNPs rather, single nucleotide polymorphism, repetitive DNA sequence, that are going to, you know, that will tell us about our variation somewhere. Okay, but here what I'm trying to say is that in DNA there are overlapping regions. There are regions which are overlapping. There are sequences which are overlapping. So basically that will be figured out, and as per, on the basis of that overlapping regions, bache, right, these sequence will be arranged. That overlapping regions will be checked and that sequence will be arranged. That is what you need to keep in your mind. Okay, so this required generation of overlapping fragments for sequencing. Alignment of these sequence was humanly not possible. So we cannot check that overlapping fragments. We cannot sequence them, right? So specialized computer based programs, they were developed for it, right? And then these sequence, they were annotated and were assigned to each chromosome, like they were marked that this particular, right, this particular sequence belongs to this chromosome, it was present here on this chromosome, that is the meaning, right? So try to understand the HGP. So from this line I can easily, if I will be the examiner, I am definitely going to ask question from this particular line, right? I am not going to ask repeated questions. If I will be the examiner, I am not going to ask you the repeated question, right? If I have to set the paper, only 30% paper will be from the, like exactly PYQs will be picked up, rest 70% will be kind of new questions, again from NCERT. Okay, again from NCERT. Are you getting my point? Are you getting my point? So I am definitely going to ask question from this, that how are you going to arrange that sequence? So you should know on the basis of overlapping regions. Okay, on the basis of overlapping region. Okay, so sequence, so what they obtained, the sequence of chromosome 1 was completed in May 2006 of the 24 human chromosome. Now you know why 24, because 22 are the normal body chromosome, and then X and Y to be sequenced. Understood? X and Y to be sequenced. Is that clear? Is that clear? So what information we got from this? Tell me, what information we got from this? Tell me. Right, what are that salient features that we got to know? That we have 3164.7 million, isn't it? Please open your NCERT. It is given in NCERT. So how many nucleotide bases do we have? How many nucleotide bases do we have? This 3164.7 million nucleotide bases. So make sure you do not make mistake here. Make sure it's million, it's not billion. Okay, make sure it's million, it's not billion. That is important. Clear? So average gene consists of 3000 bases, right? You know, remember this point, bache. Average gene consists of 3000 bases, and you know that largest human gene is, another important question, bache. Largest human gene is dystrophin gene. Largest human gene is dystrophin gene. Remember? Do you remember this information? It is dystrophin gene. This is important, and it is having how many bases? It is having 2.4 million bases. Imagine, on an average we are assuming 3000 bases, but this dystrophin is having 2.4 billion bases. How many bases are there? 2.4 billion bases are there. Is that clear? So total gene estimated, right, total gene estimated were 30,000. Earlier now we estimated near about 80,000 on 1 lakh, but total was 30,000. And you know that the smallest gene, right, that is going to be the Y chromosome. Y chromosome is having, very less genes. Y chromosome is having very less genes, right? So this, and 99.9% DNA is same, 0.1% is different. We are having the repetitive DNA sequences. All that are the salient features of the HGP. Hanji Zain bache, what's your doubt? Please tell me. Right, all that are the salient features of that HGP. Clear bache? Clear bache? And even after HGP, I am not mentioning the salient features here bache. You know that it is given in NCERT. That is your homework to study them line by line. Right, that is your homework to study them line by line. That is your homework. Clear? Right. So after this human genome project, we got to know that still the function of more than 50% of the genes, it is unknown. Okay, and we have repetitive DNA sequences, and that repetitive DNA sequences, like there are many sequences which are repeated in our case बच्चे, clear बच्चे, clear 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 clear, 100 to 1000 time, right? They are not going to code for any protein. They will not tell us, they will not give us the information for any protein, but they will tell us about the chromosome structure, the stability, the dynamics related to evolution, right? Sometimes that similarities, no doubt, all that repetitive sequences, they are different in every individual, but in the same families, kind of somewhere similarities will be there, somewhere. Clear bache? Clear bache? Is that clear? Is that clear? So that is what you people need to know. Okay, and you know that Y chromosome is having most gene, Y has the fewest, right? Chromosome 1 is having maximum genes 2968, I guess, and Y chromosome is having few, that is near about 231 or 291. What was it? Please let me know. Please check your NCERT and please let me know. 231 or 261? Right? And as I said, the scientists, they figured out one thing, bache, SNPs, right? We pronounce it as SNPs. It is SNPs. That means single nucleotide polymorphism, right? Single nucleotide polymorphism. So you know, after studying HGP, right people? After studying HGP, the scientists, they identified 1.4 million locations where single base DNA differences was there, right? They identified 1.4 million locations, right, where 5.4 million locations where single base polymorphism was there. I'll explain you this thing. It is SNPs, right? They are also responsible for the differences that we have. Let's say this is the DNA of one individual, second individual and third individual. And yes, I need to talk about something else as well. Randomly I'm picking up one sequence, okay? So look at this. Let's say this is the DNA of individual A, individual B, individual C. Okay. So here, rest everything is same. Okay. Rest everything is same. But if you will pay attention, see ATCG, CG, ATTA. ATCG, CG. So see, this is the difference. Look at this. This is the difference. Can you see that? Can you see that? Rest everything is same. Rest everything is same, but there is a difference of only one nucleotide here in these DNA. Like in my case, let's say it's C. In your case, let's say it's T. In other person's case, let's say it is A. So single nucleotide polymorphism. That is the SNPs, bache. So scientists, they identified 5.4 million location where single, right bache, where there is a single base DNA difference. Is that clear? Single base DNA difference is there. Clear bache? Clear, clear, clear. Is that clear? Sure. So this is about the salient features. So I request you people to read it from NCERT. Okay. Okay. And do you know the first prokaryote that was sequenced completely was your Haemophilus influenzae? Do you know that? First prokaryote that was sequenced was Haemophilus influenzae. Do you know that? First prokaryote in which genome was completely sequenced, it was Haemophilus influenzae. Is that clear? So this is about the HGP. If you people are having any doubt, you're free to ask me the questions. Tell me. If you want to ask something, you can tell me. Yes? So because of this human genome project, we got something else, and that we call as DNA fingerprinting, also known as DNA profiling, also known as DNA typing. So yes, you should know the another name as well. So this technique was invented by Sir Alec Jeffrey, bache, right? And in India, it was started by, it was invented by, when the word is invented by Sir Alec Jeffrey, but in India, In India, it was initiated by Dr. V.K. Kashyap and Dr. Lal Singh. Or Dr. Lal Singh. They started this. Okay. The first time they performed it, they started in India. It was started by Dr. V.K. Kashyap and Dr. Lal Singh. Clear, bache? So, see, here now, we really need to focus on NCERT. I'll tell you this thing very clearly, bache. The steps of DNA fingerprinting, we will discuss it in detail in biotechnology. I'm not going to complete this topic here, right? We will discuss that in biotechnology. Why? Because in biotechnology, you talk about PCR, you talk about gel electrophoresis. We will also discuss blotting techniques. Then you can relate that DNA fingerprinting properly. Okay, bache. Then you can relate that DNA fingerprinting properly. But here I'll just tell you about the satellite DNA so that you don't get confused. Okay, in the biotech also, wherever the DNA fingerprinting is given, there also we will discuss this. But yes, you will have better clarity of this topic in biotechnology. Okay, so remember one thing, that polymorphism in human DNA, it is the basis of that what we call it, DNA fingerprinting. Okay, so we have 99.9% DNA same. There is a difference of only 0.1%, and that difference is also because of the repetitive DNA sequences. And that repetitive DNA sequences, they are unique to human being. They are not going to form any protein. But let's say if 99.9% DNA in our case, if it is same, and if we are performing centrifugation, right? If we are performing what? We are performing centrifugation. What is going to happen in centrifugation? The things which are having mass, they will come at the base, and the lighter things will come up. This is what we know. Now let's say in our DNA, when we know the bulk DNA, the majority of DNA, 99.9% DNA is same, but there is a difference of 0.1% DNA. So obviously bulk DNA, in our case, that bulk DNA will be same. That 0.1% wala will give different peak. It will give different bands, and that bands are used to figure out the differences in two individuals. Just say we have the fingerprints. They are unique to everyone, but still, you know that we cannot totally rely on it. Now there are the techniques, right? There are the techniques by which, even by using I think sheets or something, you people see such movies, right? One can use other person's fingerprints, right? But in the case of DNA fingerprinting, it is not like that, because here that unique DNA will be checked, that bands will be studied. Okay, so DNA fingerprinting involves identifying differences in some specific region in DNA sequence, and that specific region is nothing but the repetitive DNA. So our repetitive DNA is going to differentiate us. Please consider this point, right? And in these sequences, a small stretch of DNA is repeated many times. Let's say there is in that DNA, what they said, a small stretch of DNA is repeated many times. Let's say there is a sequence GTACCG, randomly I'm telling you, that will be repeated so many times. That repetitive DNA is differentiating us. Okay, so it will be separated from bulk genomic DNA. Now can you understand? Bulk genomic DNA means that majority of DNA has a different peak during density gradient centrifugation. So when we perform density gradient centrifugation, this repetitive DNA will be different from that bulk genomic DNA, from that 99.9% same DNA. So bulk DNA, it will form major peak. Obviously you are talking about the density. This will give you a major peak, right? And other small peaks are your satellite DNA. Satellite DNA is nothing but the repetitive DNA. So depending on base composition, whether it is AT rich or GC rich, length of segment and number of repetitive units, the satellite DNA is classified into different categories. So what is the satellite DNA? It is that small peaks that we are going to obtain during density gradient centrifugation. It is because of the repeated DNA sequence. It is because of the repeated DNA sequence. Right, bache? So here, as per the composition of base, as per the length of the segment and the number We will consider the composition, the length and the number of repeated units. Accordingly, we divide satellite DNA into various categories like microsatellite and mini satellite. So that is your homework, bache. In the comment section, I want you people to write the differences in mini satellite and the microsatellite. Right, in mini satellite and the microsatellite. So they are not going to code for any protein, you know that, right? So it's just that they will show high degree of polymorphism, right? It is different. This repetitive sequence is different in different individual, and that's the basis of DNA fingerprinting. Understood? That is the basis of DNA fingerprinting. So you know that for that we should have the source of DNA. RBCs, mature RBCs cannot be the source of DNA because they do not have the nucleus. So be aware, right, when you select the material. So we need the hair, saliva, skin, bone, sperm sample, anything it can be. Okay, right. So it's a very useful identification tool in forensic application. So these polymorphism, different forms, that repetitive sequences, they are heritable. Right, they are inheritable from parents to children. So it is the basis of paternity testing also. Like my DNA, obviously it will show similarity with my parents. Okay, it is going to show similarity with my parents' DNA, right? So as polymorphism, you understand now, poly means many, morph means form. So as polymorphism is DNA sequence is the basis of polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. Another important MCQ. I still remember there was a question in NEET 2022 paper which confused many teachers, right? We missed this point. So remember, polymorphism in DNA is the basis of genetic mapping as well as DNA fingerprinting, right? Genetic mapping and DNA fingerprinting. That is why I added the NCERT, because sometimes some questions, they are so unexpected, we do not, right, we miss these points from NCERT. Okay, so polymorphism is not just the basis of DNA fingerprinting, it is the basis of genetic mapping as well. It is the basis of genetic mapping as well. Is that clear? So it is essential that we understand what DNA polymorphism means in simple terms. So poly means many, form, so variation at genetic level which is due to mutation, it is what polymorphism is. Clear, bache? And these mutations are inheritable, means they will pass from one parent to another. Okay, so in a population, obviously we observe it at a high rate. Okay, right, so probability of such variation is to be observed in non-coding DNA sequence. It will be higher as mutation in these sequence may not have an immediate effect in an individual reproductive ability. Now let's say less than 2% DNA is giving us the information for coding the protein. If that mutations are in that less than 2% DNA, that will be a problematic situation because that will affect our physiology. Now when you are talking about this non-coding DNA, about this repetitive sequences, or about this DNA which is not giving us protein, if there will be mutations there, that is not going to affect our system, that is not going to affect our physiology. Yeah, that can differentiate us from one person to another, but that is not affecting our physiology. That is the meaning of this line, right? Because our functional protein is not getting affected, right? Our functional protein is not getting affected, right? So these mutations, they will keep on accumulating generation after generation because they are in non-coding sequences and they are the basis of polymorphism. So mutation in the non-coding region which is not giving us protein, when it gets accumulated, it is the basis of variability or polymorphism, right? So there are different type of polymorphism. I told you about the SNPs, that is single nucleotide polymorphism as well. So bache, this process, yes, in detail we will be discussing it in in biotechnology, because there you will understand it properly. Okay, so these are the same points. That technique was developed by Alec Jeffrey. He used the satellite DNA as a probe and that has shown the high degree of polymorphism. He used to call it variable number tandem repeats. Okay, so he used southern blotting, radioactive ENTR. So we will discuss that part in the biotechnology. In biotechnology, it will be better clear to you all. Okay, so this is all about the molecular basis of inheritance. tomorrow at 11 a.m. there will be a special class on Unacademy platform, and in that special class, people, we will be solving the questions. So if you will be having any doubt, you can ask me there, right? We covered this chapter in 2 parts. The only reason was I really don't want to compromise with the content. I know on that day, because I was not getting good response from students, so I was thinking that you are not in the condition to grasp that much, right? Okay, so I really want you people to revise from both the parts, right? And please be sincere. See you all tomorrow at 11 a.m. for the special class. And moreover, as I said, after 3 days, this is the lecture that we have. Hit the like button, be the part of this lecture, right? If you will follow the series, see, principles done, molecular basis done, biotech, both the chapters done. So these 4 chapters, even the evolution is done. These 5 chapters are the most important chapters that we have covered, right? The most important chapters. Okay, so please start watching the classes, right? Please start watching the classes. Please start revising these topics properly. And yes, here we have the revision plan for all of you. Okay, here we have the revision plan for all of you. See, Pankuri ma'am has also posted the important topics for scoring good marks in your botany. And moreover, this is the complete strategy video, right? It is the complete strategy video, right? It is very realistic. Here I talked about the important topics, important chapters, the routine that you need to follow, and actually is it possible or not, this is what we have discussed here. Okay, so just see in the description box, there is a link that is for our batch NEET Endgame. Still, it's not too late to join that batch, right? After 2 to 3 classes, I will be starting the human physiology in that batch. In detail, we will be discussing human physiology, right, bache? Other important chapters are going on in other subjects. So please do not waste your time. Join this batch. This is the coupon code that you have to apply, and you will get this batch at a price of 7599, where there will be a test, doubt session, everything will be provided to you, right, bache? Right? And even in the description box there is a Resolve 2024 as well. Okay, so you can get this subscription for 30 months, 24 months, 18 months in different ways. Clear, bache? Okay, so we'll share the PDF in Telegram group. See you all after 3 days. Till then, please stay tuned, keep revising, and promise me that you are going to revise this chapter from NCERT. Yes, everyone, please promise me that yes ma'am, we are going to revise this chapter from NCERT right after the class, right? Today you are free, so right after the class you are going to revise it. And I want you people to write a comment, right? You liked the session or not? Okay, I really want to know that. Even if there is like, nahi ma'am, we don't like it, just put it in comment section. Okay, done? So we'll share the PDF in Telegram group. So take care. Thank you so much. Stay tuned. Bye bye.